Good afternoon. My name is Dr. Ruby Mehta, and I am a pediatric hepatologist and acting team leader in the Division of Hepatology and Nutrition. I will be moderating the afternoon session today. The second group of speakers will walk us through the metabolism of phytosterol and role of phytosterol in development of PNALD or IFALD. The first speaker for the afternoon is Dr. Douglas Burrin. He's currently appointed as a research physiologist with the USDA ARS and professor of pediatrics at Baylor College of Medicine. Dr. Burrin is also a director of fellowship research training in the Division of Gastroenterology, Hepatology, Nutrition at Baylor College of Medicine. He also serves in the leadership of NIDDK-funded Texas Medical Center Digestive Disease Center and as a director, pilot, and feasibility program director. Dr. Burrin will be discussing the metabolism of phytosterols and non-clinical evidence supporting the association between plant-based ILEs and PNALD and IFALD.
So I want to thank the organizers, Judy Recruson and others, for the opportunity to speak today about some of our preclinical work. The title of my talk is Metabolism of Phytosterols, Preclinical Evidence Associating Plant-Based Lipid Emulsions and PNALD. First, I'll give a brief outline of my talk today. I'm going to talk a little bit about phytosterol chemistry and physiology. I'm then going to tell you a little bit about our neonatal piglet model of pediatric cholestatic disease. And then I'm going to briefly give you some perspective about phytosterolemia in infants and children from the literature, and then spend most of the talk talking about preclinical evidence of cholestasis and phytosterolemia in our TPN-fed pig studies that we've published recently, and then finally close with the mechanisms for how we think phytosterols are disrupting hepatic bile acid metabolism. So first I want to give you a little bit of background on the chemistry of phytosterols. So shown on this slide in the top left is the structure of cholesterol, which is the 5-ring structure for cholesterol that is present in mammalian cell membranes, and it serves as a precursor for bile acid synthesis in mammals. And this occurs through hydroxylation at the 7 position of the ring structure. And this is accomplished by the rate-limiting enzyme in hepatic cells, in hepatocytes, which is CYP7A1. Also shown on the bottom are similar structures for plant-based phytosterols. As you can see, they're very similar in structure to cholesterol and bile acids, and there are differences in terms of the type of phytosterols is mainly based on the side chain structure associated with each of the phytosterols. This slide shows a little bit about the sterol physiology and transport. And what I've done is to show you for both cholesterol and phytosterols how they are transported through the gut and the liver. And what I'm showing you here are the ATP binding cassette transporters G5 and G8, as well as the Niemann-Pick C1-like transporter, both which are expressed in the gut. So under normal circumstances in adults, there's a quite large range in phytosterol intake, which can be anywhere between 100 and 400 milligrams per day. These phytosterols, as well as cholesterol, are ingested, they're absorbed into the gastrointestinal tract lumen, and then they are transported into the epithelium, first through this Niemann-Pick C1-like transporter. They go into the epithelial enterocytes, and then in the case of phytosterols, much of this is then retransported and exported out into the lumen of the gut. And so under normal circumstances, there's a very low absorption of phytosterols ingested in the diet, somewhere around 5%. But a larger proportion of the cholesterol is absorbed from the epithelium of the gut into the lymphatics and into the circulation. Once into the circulation, cholesterol, as well as any phytosterols that are absorbed into the bloodstream, are then re-excreted out through the biliary flow, through the ABCG5G8 heterodimeric transporters that are expressed on hepatocytes. These phytosterols and cholesterol then go into the gut and they are then excreted from the body. Under conditions of IV lipid administration, or TPN, under conditions in infants in this case where the phytosterol intake might range between 1 and 5 or upwards of 10 milligrams per day, these phytosterols bypass the enteral route of absorption and they go directly into the bloodstream. They are then taken up by the liver, and then under normal conditions where there's normal biliary flow, these phytosterols would be transported out of the liver into the biliary system through the ABCG5 transporters and then excreted in the gut through the normal route. However, under conditions of cholestasis, as we'll talk about today, phytosterols accumulate in the liver because these transporters are downregulated for various reasons of which I'll talk about. And so what happens is there's a limited excretion of phytosterols under conditions of phytosterolemia, and they accumulate both in the liver and in the blood. So what I'm going to show you in the next few slides are some data to give you perspective of the phytosterol concentrations in infants and children. I'm going to start with data that was reported in the seminal paper from Clayton and others that was published in Gastroenterology in 1993. And what they did was report concentrations of phytosterols shown here in total phytosterols, which include most of the principal phytosterols of camptosterol, stigmasterol, and cytosterol. They range somewhere in a neighborhood between 20 and 30 micromolar under normal control conditions where there's normal liver function. However, in infants that are receiving parenteral nutrition, either for short or longer periods of time, when there is a mild liver dysfunction, the phytosterol concentrations can get up in the hundreds, in this case over 200, and in very severe liver dysfunction noted in children, the phytosterol concentrations can get as high as over 1,000 and almost 1,500. So also in this study from Clayton and others, they showed an important concept, and it's shown here, and that is when the phytosterol lipid load is increased, shown on the left-hand panel, there is then an eventual increase in phytosterol concentrations in the plasma, which you can see at about day 50 in the center panel. But when phytosterol loads are decreased, as you can see on the left, this then results in a decrease in plasma phytosterols. And what you can see in the final panel on the right is that as phytosterols increase, there's a direct correlation with the concentration of bilirubin in the blood. This shows data from a more recent study in preterm infants published in 2015, where preterm infants were studied at about 32 weeks of gestation, and these infants were receiving 2 to 3 grams of a soy oil lipid emulsion starting at birth. And you can see over the first week of life, the 3 principal phytosterols shown here in micromolar for cytosterol, camptosterol, and stigmasterol increase with time, particularly for cyto and camptosterol. And what's important to realize is these concentrations increase in the range somewhere between 25 and 75 micromolar. And so this will be important as we look at some of the data from our pig studies. So to give you an overview of our preterm and term pig model, I'm showing you here really a conceptual diagram on the right, and that is how we think about using the pig as a model to study issues of nutrition both in healthy term and preterm infants, but also in infants that experience disease, in this case like necrotizing enterocolitis or TPN. And we really feel like the pig is has an advantage over the mouse, which is the dominant rodent or animal model in biomedicine, and that pigs are large enough at birth and they can be delivered premature. And this makes things very feasible to do, in this case, like give parenteral nutrition, or in our case, we also give enteral nutrition support. So what's important to know about our pig model, and the study is going to talk about today, is the lipid load that we give to pigs is 2 to 3 times higher than one is normally give to a human infant. And this is because the pig has a much higher nutrient requirement for energy and protein. And under most of the studies that I'm going to talk about today, our studies that have been 2 to 3 weeks in length result in what we consider to be a mild cholestasis, but we do have in some cases evidence of injury from cholangitis, steatosis, and inflammation. So I'm going to talk about in this presentation about 3 different lipid emulsions that most of you are very familiar with. And the first is intralipid, which has been approved for decades. It is a pure soybean oil-based lipid emulsion that contains enriched phytosterols and N6-PUFAs. Also Omegaven, which is now approved, which is a fish oil-based emulsion which does not contain phytosterols and is enriched in DHA and EPA. And then finally SMOF-lipid, which is a multi-component lipid emulsion composed of these 4 different oils. It does contain phytosterols and was approved recently. So when we began to study these emulsions in our pig model, we really wanted to ask a basic question, and that is, do new generation lipid emulsions prevent PNALD, and what is the mechanism? So the first study we did was done by an MD Ph.D. student, Hester Vardingenbrunk, and Ken Ng, who is a PDGI fellow. And we used pre-trim pigs, and we studied these 3 main lipid emulsions that I've outlined, as well as an enteral group that received enteral milk-based formula. These pigs were given a lipid intake of 5 grams per kilo per day. The study lasted for 2 weeks, and we assessed liver function and histopathology. And shown on this slide, you can see are the serum levels of total bilirubin and GGT. And what you can appreciate in the top left is that after 14 days, those pigs that received intralipid demonstrated a marked increase in bilirubin and in GGT. But you can see in the Omegaven and the SMOF groups that this tended to be prevented or was lower compared to intralipid. We also showed that intralipid resulted in an increase in serum bile acids that tended to be lower with Omegaven and SMOF. And this was also found in the case of the liver bile acid content after 14 days. Shown on this slide are some histopathological evidence of liver injury. Shown in the bottom right is a normal H&E of a liver from a healthy pig, and then you can see in the top left evidence of pericholangitis, top right acute cholangitis and ballooning around the portal tracts. And then also on the left, lower left, you can see portal leukocyte infiltration. And we also notice frequently in pigs that experience cholestasis, especially with intralipid, is that there's a yellowish color of the liver that we think occurs due to the accumulation of bilirubin and bile acids and fat. And in the case of fat, we do experience and see evidence of steatosis. On the top here, you can see the liver triglyceride chemical composition, which is elevated almost 3-fold with intralipid, and this is prevented with either Omegaven and SMOF. And on the bottom, you can see red O staining that gives qualitative evidence of steatosis with intralipid. So I'm going to talk now about the phytosterol concentrations, and I just wanted to give you some evidence of the concentration of phytosterols in intralipid. These have been reported widely in the literature and different studies and papers. In this case, we analyzed intralipid to have about 900 micromoles total. If you include beta-sitosterol, camptosterol, and stigmasterol, you can see that SMOF is substantially lower than intralipid, and Omegaven does not contain, and we could not detect phytosterols in Omegaven. And in the case of the enteral formula that we fed in this study, we did detect some low level of phytosterols in the formula. So shown on this slide is the daily phytosterol intake, and you can see that it amounts to, in the case of intralipid, somewhere around 20 micromoles per kilo per day. There's very low or no intake of phytosterol in the Omegaven group, and then a substantially lower by approximately half phytosterol intake in the SMOF lipid group. And what you can see is this is basically reflected in the plasma phytosterol concentration and in the liver phytosterol concentration shown on the right. We then followed up this study and we wanted to look at a more long-term study. We also looked at the effect of intralipid and SMOF, and then we studied a third emulsion that was an experimental emulsion that we were examining in collaboration with Fresenius that contained higher amounts of DHA, arachidonic acid, and lower EPA. This study was conducted by a graduate student in the lab, Lee Call, and a neonatology fellow, Tiffany Molina Rubenstein. And this was a 3-week study with a similar lipid intake of 5 grams per kilo per day. And we assessed similar outcomes. But in this case, we specifically looked at bile acid pools. And first, I just want to show you evidence of the direct bilirubin levels that we showed. Again, pigs that are given intralipid, there's a marked increase in direct bilirubin between 14 and 22 days that is generally prevented by SMOF in the experimental SMOF-like emulsion. And this also was evident in plasma levels of GGT being highest with intralipid. So in this slide, what I'm trying to show you is evidence of changes in the hepatic and the gut pool sizes for bile acids. And I've expressed this as total bile acid pool size in micromole per kilo per unit of body weight. And what you can see is in the liver, in all 3 groups that received the lipid emulsions through TPN, there's a significant increase, almost doubling of the liver bile acid pool. But when you look at the gallbladder and the gut components, you can see that those pigs that receive intralipid had substantially lower pools of bile acids compared to the intro group, as well as either the SMOF or experimental group. And what is remarkable in this study is that the pool of bile acids that appears in the gallbladder and in the gut contents, which is evidence of bile flow, is essentially maintained in the SMOF lipid group and the experimental group, and it's almost identical to what we found in the intro group. So we think this provided really strong evidence that even under conditions where there's no enteral feeding and you're providing a new generation lipid emulsion, there continues to be normal bile flow. Shown on this slide is evidence that not only bile, but the phytosterols that we know are excreted in bile through the liver are maintained with new generation emulsions, in this case SMOF. Shown here, you can see the accumulation of plasma phytosterols with intralipid that are prevented with either SMOF or the experimental emulsion. And you can see that happens in the liver pool. Actually, of phytosterols tends to be lower in SMOF and experimental compared to the intralipid group. But when you look at the gallbladder pool of phytosterols, there was a much higher excretion and concentration of phytosterols in the bile in the SMOF in the experimental group, consistent with the maintenance of bile flow and the excretion of phytosterols into bile, which prevents their accumulation in the liver and in the blood. So finally, I just wanted to outline a study that we reported recently in collaboration with Fresenius, and this was a study that was conducted by Greg Guthrie, who's been involved in in all of the studies that I've talked about today. And the aims of the study were really twofold. First, we wanted to manipulate the phytosterol content without changing the lipid emulsion composition, because all the previous studies I've shown you today, we had differences in lipid composition, so it's confounded by that. We also wanted to produce a range of phytosterols that were between 0 and 150% of normal that you might find in a soy-based emulsion like intralipid, shown graphically on this illustration here. So we had 4 groups. We had an enteral formula group. We had a depleted group where we tried to deplete phytosterols from a soy-based emulsion, in this case intralipid. We had normal phytosterols that were representative of intralipid. And then we had an enriched group where we added back the extracted phytosterols that we took out from the soy-based emulsion. This is a study that was also done in preterm pigs for 21 days at a lipid load of 5 grams per kilo per day. So before I show you the data, I wanted to give you some evidence of how well we did in terms of changing and modifying the lipid emulsion. So on this table, you can see on the top the 3 dominant phytosterols being beta-sitosterol, camptosterol, and stigmasterol. And what you can see is that these 3 phytosterols represent somewhere between 80 to 90% of all the phytosterols that we measured in these different emulsions. So the total phytosterol shown here on the bottom range somewhere between 300 micromoles per liter up to 1800. So we were not able to completely deplete phytosterols from the soy-based emulsion, and this I think is one of the challenges going forward with this approach. The concentration was quite high in the intralipid group. It was around 1280 micromolar, and then it was about 144% of control in the enriched group. Another important thing to note is that the cholesterol content of the emulsions was not substantially different, about 10% higher in the enriched group. So if we look first at the plasma phytosterol concentration shown here in the top left, what you can see is both the intralipid and the enriched group had an increase in phytosterols that began really at day 7 and became more marked after 3 weeks. At the same time, you can see a correlating increase in serum direct bilirubin, especially in the enriched group, but at day 7, at day 21, also remarkably in the intralipid group, consistent with some of our previous work with intralipid. So we tried to correlate this, and shown on the bottom 2 panels on the left, it shows the data from this particular study where we correlated plasma phytosterol concentration in the 4 groups with their direct bilirubin levels, and you can see there's a fairly strong Spearman correlation coefficient that's highly significant, suggesting an increase in bilirubin with plasma phytosterols. And then shown on the right, what I've done is to take all 3 of the studies I've shown you today, and they are color-coded here, and we found a direct correlation, again, with a fairly substantial R value, and that was highly significant, suggesting that as plasma phytosterols increase, markers of cholestasis at least increase directly and linearly. This slide also shows that under conditions of phytosterolemia, shown in the top left here, which is the plasma bile acid concentrations that also increase with the plasma phytosterol concentrations, you get an accumulation of bile acids with an enrichment of phytosterols in the liver. This suggests that as phytosterols lead to cholestasis, there's an accumulation of bile acids in the liver, and there's a reduction in the concentration of bile that appears in the gallbladder, shown on the top or on the bottom left, and also in the gut contents. So we think this shows good evidence that phytosterolemia leads to cholestasis and reduced bile flow in the intestine, which is what you would predict. Finally, I want to just give some comments and some ideas about how we think phytosterols are inducing cholestasis and liver injury. And we began these studies really focusing on the main bile acid sensor, which is the nuclear receptor, the farnesoid X receptor that's expressed in the liver and in the gut. And FXR, under normal circumstances, is activated by bile acids. And once FXR is activated, it then triggers the upregulation of target genes, most importantly, the bile salt export pump, which pumps bile acids out of the hepatocyte into the biliary system, and it also upregulates the ABCG5 and G8 transporters, which transport sterols out of the hepatocyte into bile. Importantly, also, FXR activation suppresses CYP7A1, which is, again, the rate-limiting enzyme in bile acid synthesis. So to come back to the previous study that I showed you, when we looked at the expression of these genes in hepatic liver tissue, what you can see is that in all of the pigs that receive parenteral nutrition, regardless of whether it was intralipid, omega-9 or SMOF, there's a substantial reduction in FXR, in CYP7A1 messenger RNA, and also importantly BSEP messenger RNA, which is consistent with the idea that then bile acids would accumulate in the liver and in the hepatocyte. We also showed in a study that was consistent with previous data from Beth Carter and Saul Karpin in mouse and human hepatocytes that when you stimulate hepatocytes with bile acids, in this case, we stimulated pig hepatocytes with the FXR agonist of beta-cholic acid. And what we found is that there's a marked upregulation of BSEP, the transporter for bile acids. But when you co-incubate these hepatocytes with bile acids and with phytosterols, there's a blunting of BSEP expression, suggesting on the right that phytosterols antagonize the effect of FXR. So finally, what I'm showing you on this slide is really a summation of a number of very elegant studies that were done by Karim El-Kazmi and others in Ron Sokol's lab that have been published over the last several years. And what they have shown, first off, is to show that under conditions of TPN, there's a leaky gut that many of you are aware of that leads to increased permeability to endotoxin from the gut that then seeps into the liver, and endotoxin activates the TLR4 receptor in Kupffer cells within the liver. We also know that phytosterols can directly activate Kupffer cells, even in the presence or absence of endotoxin. And under both conditions, when these Kupffer cells are activated, they secrete the pro-inflammatory cytokine IL-1 beta. And what these studies showed is that IL-1 beta that then is released from Kupffer cells in a paracrine fashion activates NF-κB in the hepatocyte and then stimulates and trans-represses nuclear receptors in the hepatocyte, namely FXR, which I've talked about, but also LXR, the liver X receptor, and the other nuclear receptor, LRH1. And together, these nuclear receptors participate in the activation of important transporters that pump bile acids out of the hepatocyte into bile and phytosterols out of the hepatocyte into bile. So when these are suppressed by IL-1 beta activation, this downregulates these transporters and leads to accumulation of phytosterols and bile acids in the hepatocyte and a reduction in their excretion in the bile. We also know that the accumulation of phytosterols, as I showed on the previous slide in our own work, antagonizes the ability of bile acids to then activate BSEP. And so taken together, this shows what I think is a reasonable unifying model that shows how phytosterols and inflammation associated with TPN leads to cholestasis and phytosterolemia. So finally, I just want to acknowledge the people that were involved in these studies, especially Barbara Stoll and also Greg Guthrie, who was involved in a number of these studies, and particularly the last study, but also Li Wei, Lee Call, and Tiffany, and a number of people that involved in the group at Baylor, but also Hans van Houdever and Hester involved in the studies early on. And I want to thank the Texas Medical Center Digestive Disease Center and support from both USDA and the NIDDK.
Thank you, Dr. Burrin. Our next speaker is Dr. Kishore Iyer. He's Director of Adult and Pediatric Intestinal Rehabilitation and Transplantation at Mount Sinai Medical Center in New York. And he's also Professor of Transplant Surgery and Pediatrics at the Icahn School of Medicine at Mount Sinai in New York. He will be discussing the clinical evidence supporting the association between plant-based ILEs and PNALD and IFALD.
Good afternoon. I want to thank the organizers at the FDA for inviting me to this workshop and for asking me to speak on the clinical evidence supporting the association between plant-based intravenous lipid emulsions and parenteral nutrition-associated liver disease, otherwise referred to, I think erroneously, as intestinal failure-associated liver disease. Here are my disclosures. None are relevant to the current presentation. To the extent possible, I will address only evidence-based science in my presentation. Parenteral nutrition-associated liver disease is a syndrome of progressive cholestatic liver disease associated with parenteral nutrition in the setting of intestinal failure. Implicit in this definition is the absence of any other known primary liver disease. This syndrome is recognized to be multifactorial in origin, with the various factors shown there being important, particularly sepsis and prematurity. I put this slide up for two interesting reasons. One, some of you may have noticed the slightly grainy texture of this slide, and indeed, this was originally a 35 mm glass slide that I made in 1993 and then transferred to PowerPoint. And the other is because when Frank Sinatra from Los Angeles, before you jump to the wrong conclusions, not the singer from Children's Hospital of Los Angeles, when he first reported this model, it feels like nothing has changed over the 40 years since the model was first reported. Central to the idea of parenteral nutrition-associated cholestasis is a reduction in bile flow, which is ultimately the pathophysiologic definition of cholestasis. We've already discussed some of the risk factors for this syndrome that lead to parenteral nutrition-associated cholestasis. I put up these three historic papers in the hope that this may jog something in your thinking. Most importantly, to say that the earliest parenteral nutrition solutions contained no fat emulsion. In fact, the early lipid emulsion, experimental cottonseed oil, was hastily withdrawn due to pulmonary toxicities, not to do with the liver, in the 60s. During this period, there were sporadic reports of mild liver dysfunction. In fact, the earliest clinical report that I can find was a letter to the editor in Journal of Pediatrics from Pedin reporting liver dysfunction in a child on PN. There were indeed sporadic reports of mild liver dysfunction in this period, but it was only after the introduction of soy-based lipid emulsion in the early 70s, understandably as a concentrated caloric source and for the prevention of essential fatty acid deficiency, that there was a concurrent rise in progressive and often fatal parenteral nutrition-associated liver disease. The first clinical series of patients that described a relation between fat emulsion injection and parenteral nutrition-associated cholestasis came from Allardyce in the now defunct journal Surgery, Gynecology and Obstetrics, I guess now rebranded as the World Journal of Surgery. Allardyce reported on 35 men receiving PN for more than 3 weeks. He showed that there was progressive cholestasis in 10 of the 18 men receiving 3 grams per kilo per day of fat. In contrast, cholestasis was observed only in 1 of 17 patients receiving 1 gram per kilo per day of fat. There appeared to be no influence of the amount or type of dextrose or amino acid calories. There was an improvement in cholestasis with discontinuing parenteral nutrition or, in fact, simply by reducing the dose of lipid emulsion. However, no causal relationships were reported in this study. Peter Clayton, working at the Hospital for Sick Children at Great Ormond Street in London and the Institute of Child Health, made a very clever, if serendipitous, observation that led him to test the hypothesis that phytosterols in lipid emulsions can accumulate in serum of patients on parenteral nutrition and contribute to cholestatic liver disease. Peter and colleagues studied 29 children aged 2 months to 9 years on long-term parenteral nutrition. 5 of the 29 children had severe parenteral nutrition-associated cholestasis, meeting really any definition of severe liver disease that we would accept today, with 24 of the 29 children having mostly mild and a few moderate cases of parenteral nutrition-associated liver disease. Peter and colleagues also studied 29 age-matched controls not receiving parenteral nutrition. 5 of the 29 had congenital cholestatic liver disease, non-PN, such as biliary atresia. The investigators correlated levels of plant sterols in serum with LFTs. I will point out here that though we loosely use the term LFTs, while admittedly measurement of serum bilirubin measures excretory liver function, we should acknowledge that elevated levels of AST and ALT do not actually reflect liver function. They simply reflect hepatocellular injury, but we will stick with convention and talk about LFTs in this context as well. So what are these phytosterols? Phytosterols are plant sterols present in large amounts in intralipid and other soy-based lipid emulsions. As you can see in the biochemical structure below, the nucleus, the sterol nucleus is identical to that seen in cholesterol. They differ by virtue of possessing alkylated side chains. Beta-cytosterol, the prototype plant sterol shown here, has an ethyl side chain as shown there. Of interest, the routine enzymatic assay for measurement of serum cholesterol works by cleaving the hydroxyl group in the 7 position of the sterol nucleus, and thus, as you can imagine, is unable to distinguish between cholesterol or any of the plant sterols in serum. I find it interesting that many of the early reports of parenteral nutrition-associated cholestasis report high levels of cholesterol in serum, though measured with the conventional cholesterol 7-alpha hydroxylase enzymatic assay. Clayton and colleagues reported progressive accumulation of phytosterols in serum of patients on parenteral nutrition. The plasma phytosterols rose before any change in the liver tests. The serum liver tests improved in 4 of the patients with severe liver dysfunction when plasma phytosterol levels fell in response to lowering the dose of intralipid. There were low to undetectable levels of plant sterols in controls, including, in fact, in the patients with cholestasis unrelated to PN, such as biliary atresia. This slide summarizes the results from Peter Clayton's study. The orange bar on the left shows the phytosterol concentration in pure intralipid, measured as we did in the lab as the sum of beta-cytosterol, campesterol, stigmasterol, fucosterol, and isofucosterol. What you can see on the extreme right, in patients with severe liver dysfunction, the phytosterol concentration rose in serum and, in fact, in many cases, exceeded the level seen in pure intralipid. The barely visible middle bar shows the levels of phytosterols in serum of patients with normal liver function, which was consistent with that seen in normal children and barely detectable in some cases. Not shown in this slide is that patients on PN with normal liver function, and in fact healthy controls, or patients with mild liver dysfunction, had a range of serum phytosterol concentrations between the 2 extremes. Clayton and colleagues also reported on the levels of AST, bilirubin, and platelet counts, noting that thrombocytopenia has often been reported in patients receiving intralipid. And they attempted to correlate this with the intralipid infusion rate. What you can hopefully make out on the table is that the patients with severe liver dysfunction received much higher rates of intralipid than patients with mild liver dysfunction. The graphic on the right shows the time course of phytosterolemia and hyperbilirubinemia in an infant receiving 20% intralipid. Panel A is the intralipid infusion rate, first at baseline and then lowered in response to the occurrence of a higher level of phytosterol seen in the middle panel, the middle bar of the middle panel. And you will note that the serum bilirubin concentration shown in the lower panel was initially low. With rising intralipid infusion rate, there appeared to be a rise in phytosterol concentration, and the serum bilirubin concentration in this child appeared to parallel the phytosterol concentration in serum. And in fact, when the phytosterol concentration fell in response to lowering and ultimately stopping the intralipid infusion, the bilirubin normalized. It was about this time that I had the privilege of working with Peter Clayton in his lab for the subsequent 3 or 4 years. And one of the key questions that we were confronted with was the question that Peter Clayton's study had not answered, which was the occurrence of phytosterolemia in these patients with cholestasis simply an epiphenomenon, i.e., the accumulation of phytosterols in a cholestatic child who could no longer excrete the phytosterols, or did, in fact, the phytosterol accumulation have any pathologic significance? I know my mandate was to focus on clinical studies, but I hope you will agree that the next few slides are very relevant to the clinical discussion. So the upper panel shows the gas chromatogram in a child that I encountered for the first time in 1993, who sadly ultimately died of severe parenteral nutrition-associated liver disease. What you will see there is that the plant sterols shown by the peaks of cytosterol and campesterol, the smaller peaks are stigmasterol and isofucosterol. The serum phytosterol concentrations in this child, in fact, exceeded that of cholesterol. What we were interested in doing was to create an animal model, not of DPN-associated cholestasis, but rather of parenteral nutrition-associated phytosterolemia. It occurred to us that we had to be able to deliver a phytosterol concentration, ideally in an inert solvent, as a daily dose of phytosterols alone in the absence of any other component of TPN. And we did this by creating a phytosterol mixture in an inert solvent, beta-hydroxypropyl cyclodextrin. And you can see the gas chromatogram of this solution, in fact, actually mirrors that found in pure intralipid. Running through some of our results very quickly, what we were able to show in a newborn piglet model was that with daily injection of a mixture of phytosterols as found in intralipid, in the absence of any other component of TPN, we could show a dose-related accumulation of phytosterols in serum within the 7-day period of study. There was also an accumulation of phytosterols in the liver, even though the dose relation was less obvious. And interestingly, this finding has now just recently been shown in humans. And in our terminal studies at 7 days, it appeared that the steroid-treated piglets had inhibition of bile flow and bile acid excretion, i.e. cholestasis. Interestingly, while these piglets did not become jaundiced or hyperbilirubinemia, within the 7-day period of study, we could show elevation of serum bile acids, suggesting early cholestasis. And I will emphasize that this occurred in healthy chow-fed piglets who received no other component of PN other than a mixture of phytosterols. In a related, elegant cellular model of canalicular bile acid transport, the isolated rat hepatocyte couplets, we were able to show that steroid treatment of isolated hepatocyte couplets caused a linear and dose-dependent inhibition of bile acid transport in the absence of any other component of PN. So, are there additional clinical correlates between intravenous lipid emulsion and the occurrence of parenteral nutrition-associated liver disease? Virginie Colombe in Paris reported on 10 children on long-term parenteral nutrition in 2000. These children suffered 24 episodes of cholestasis. 17 of the 24 episodes resolved with decreasing or discontinuing the dose of lipid. Marianne Cavici from Bernard Messing's group in Paris reported on 90 adult patients with permanent intestinal failure. In a multivariable model, they showed that the predictors of parenteral nutrition-associated liver disease in this cohort were the duration of PN, were a bowel remnant shorter than 50 centimeters, and finally, a dose of lipid greater than 1 gram per kilo per day. This graph shows the probability of being free of liver complications in their multivariable model. The dotted line shows that patients receiving a dose of lipid of greater than 1 gram per kg per day were conferred a relative risk of 3.4 to develop complicated parenteral nutrition-associated liver disease. This was a recent elegant study from Mikko Parakinen's group in Finland. This group has done a lot of work in this area, and they reported on 50 patients on PN, 16 of whom continued on PN while 34 had been weaned off, compared to 86 healthy controls undergoing unrelated pediatric surgery. They had liver biopsies on 40 patients. The panel on the left shows the concentration of stigmasterol, upper panel, Phytosterol in the lower panel, expressed as a ratio to the concentration of cholesterol in serum, had a linear correlation with the serum bilirubin concentration. In other words, stigmasterol concentration and Phytosterol concentration independently appear to correlate with the occurrence of hyperbilirubinemia in children on PN. The right panel shows, even more interestingly, that stigmasterol concentration, again expressed as a ratio to the concentration of serum cholesterol, correlated strongly with the presence of portal inflammation in the upper panel, and in the lower panel, Phytosterol, again similarly expressed as a ratio to the concentration of cholesterol, correlated strongly with the occurrence of cholestasis. I believe the study provides compelling evidence that the occurrence of phytosterolemia in patients of parenteral nutrition has pathologic significance, correlating, as shown in the left panel, with the occurrence of hyperbilirubinemia, and in fact, for the first time, also shown here, correlating with adverse histology in patients developing parenteral nutrition-associated liver disease. I have to take a minute to recognize my good friend, the late Dan Teitelbaum, who's done remarkable work in this area, whose many contributions, unfortunately, were prematurely cut short. Patria Kober is here today. Drs. Kober and Teitelbaum looked at the effect of lipid dose restriction in patients on parenteral nutrition. They compared 31 historic controls with a group of patients who were subjected to IV fat emulsion reduction. And what they were able to show, as you can see in the bottom right panel of this graph, is a decrease in total bilirubin in the lipid reduction group, albeit with mild essential fatty acid deficiency in some patients. This elegant slide, shared with me by Paul Wales from Toronto, tells us that there are many different ways to skin the cat of parenteral nutrition-associated liver disease. I have quite intentionally not discussed omega-1 upper left panel in this talk. It is clear that omega-1 has really moved the needle, both in terms of understanding and, in fact, in firing the conversation on lipid emulsions and TPN liver disease, but also perhaps in changing the trajectory of liver disease for many patients. What is not clear to me is whether the impact of omega-1 can be shown independent of a reduced dose of daily lipid emulsion. Nevertheless, the different investigators shown here have accomplished improvements in hyperbilirubinemia, taking several different approaches to lipid management. Finally, this slide is a reminder. This was a child I first looked after in 2000. As you can tell, this child had relatively advanced parenteral nutrition-associated liver disease, taking a multi-strategy, a multifaceted approach, including very aggressive lipid restriction. We were ultimately able to salvage his native liver, and I'm happy to say he's alive and well with his native liver. Thank you for the privilege of the floor. I look forward to discussions.
Thank you, Dr. Iyer. The next speaker is Dr. David Driscoll. He's a clinical pharmacist and has earned his Ph.D. in pharmaceutical chemistry. He will be discussing scientific challenges to producing an ILE with reduced phytosterol content.
Good afternoon. I will present the scientific challenges relating to phytosterols and plant-based triglycerides, but I will also highlight other potentially clinically important issues with the relevant triglyceride oils. For complete disclosure, I am an inventor of several patents related to omega-3 fatty acids for injection. Let's begin with a comparison of the typical phytosterol concentrations for the various plant-based oils used in lipid injectable emulsions. Of these, only soybean oil is employed alone, while the others are present as physical mixtures. The reported concentrations of phytosterol contents can vary depending on the reference source as well as where the plants are grown around the world. But clearly, soybean oil is highest. As expected, 100% soybean oil injectable emulsion has the highest concentration of phytosterols, and by gradually decreasing the soybean oil fraction of the dispersed phase of the emulsion, they are proportionately reduced. This plot illustrates a corresponding decrease in the concentration of phytosterols, coincident with reduced fractions of soybean oil highlighted by the red circles. The graph is expressed as phytosterols in micrograms per mil versus lipid emulsion. Although clinolipid only contains 20% soybean oil, it is not included in the comparison since it also contains 80% olive oil, which is also high in phytosterols. Also of note, negligible amounts, that is less than 10 micrograms per mil, are found in pure fish oil or 100% FO injectable emulsion. However, when comparing the phytosterol concentrations of commercially available soybean oil only injectable emulsions, they can vary by as much as 50%, depending upon the manufacturer. What does this mean? How consistent is the raw material source, and what other critical quality attributes of soybean oil should be considered? I should mention that these samples were taken from a single batch, but nonetheless, the variations are striking. What exactly are critical quality attributes, and how are they determined? In this slide, and according to the FDA, each pharmaceutical product must identify critical quality attributes consistent with its intended use and composition. The focus is on potentially clinically important aspects that may be influenced by emulsion quality, such as physical and chemical stability, safety, and efficacy. As phytosterols have already been identified as possibly affecting product safety, what other critical quality attributes are relevant? Alpha-linolenic acid is one of the two essential fatty acid precursors affecting efficacy, at least in terms of preventing essential fatty acid deficiency in susceptible patients. Hence, using alpha-linolenic acid as a marker or critical quality attribute, its concentration in soybean oil is important. This plot shows that soybean oils harvested in the northern hemisphere have, on average, about 15% higher concentrations of alpha-linolenic acid compared to those grown in the southern hemisphere. This variation appears to determine the product specifications of soybean oil-based lipid emulsions, as shown in the lower right-hand corner of the slide in the red box. Hence, is the variability observed in the concentrations of phytosterol, as well as the alpha-linolenic acid in soybean oil, reflective of differences in other critical quality attributes? Triglyceride oils, and especially those high in polyunsaturated fatty acids, such as soybean oil and fish oil, are most susceptible to oxidative degradation. The oxidation status of a triglyceride oil is an important critical quality attribute affecting the chemical stability of the oil that must be monitored throughout the life cycle of the product, starting with the raw material itself. This includes from the time of opening the original container or drum of oil, as well as during in-process manufacturing and after production, because oxidation is an ongoing destructive event. This slide depicts the total oxidation or TOTOX value for a set of batches from two different raw material suppliers of soybean oil. The TOTOX value combines the results from testing for the primary oxidation products via the peroxide value, or PV, that includes hydroperoxides and free fatty acids, as well as from the results obtained from testing for secondary oxidation products via the anisodine value, or AV, that includes aldehydes and ketones. Therefore, the TOTOX value assesses the quality of a fixed oil, such as soybean or fish, before, during, and after production, including shelf life determinations. Clearly, applying the TOTOX value as a critical quality attribute shows these two sources of soybean oil are not pharmaceutically equivalent. In USP Chapter 401, concerning fixed oils, for example, testing for oxidation products has been added in this general chapter to assess the oxidation state. About 10 years ago, in an attempt to update quality standards of existing USP articles, efforts have been undertaken by the USP, along with the FDA, for modernization of existing monographs, recognizing that outdated or inadequate analytical tests need to be replaced with more sensitive methods. For any fixed oil and lipid injectable emulsions, specific limits for the various critical quality attributes should be considered for each official oil monograph. Primary and secondary oxidation products affect the chemical stability of the oil as well as the possible safety of the emulsion upon infusion, since they can promote oxidative stress. In addition to soybean oil, there are also differences in the quality attributes of fish oil with respect to the omega-3 fatty acid concentrations. For example, there are two separate monographs in the European Pharmacopoeia for fish oil, and as shown here, they have very different fatty acid profiles, yet they are both used in commercial lipid injectable emulsion products. If omega-3 fatty acids are indicated to be of clinical relevance in pediatric patients with parenteral nutrition-associated liver disease, these differences might have efficacy implications. For a deeper dive into these differences, let's look at their respective chromatographs from the European Pharmacopoeia. This slide depicts the main peaks for each monograph, showing the different profiles for the top 6 fatty acids, reported as the weight percent of the fatty acid profile for each fish oil monograph. Consequently, the monographs have different specifications for the minimum concentrations of omega-3 fatty acids. Of note, when expressed as the sum of EPA and DHA, the pharmacopoeia requirement for European Pharmacopoeia monograph 1352 is about 2 times higher than European Pharmacopoeia monograph number 1912. In this slide, the focus is on the concentrations of the omega-6 fatty acids present. Although the amounts of linoleic acid in fish oil are small compared to soybean oil, fish oil is unique in that it also contains significant amounts of arachidonic acid. So how much arachidonic acid is present in currently available fish oil containing lipid injectable emulsions? Before we get into these details, however, it is important to recognize the difference between essential fatty acids that are precursors to the bioactive end products they form, as described in this slide. Omega-Avin 10% is a 100% fish oil emulsion with the omega-6 fatty acid content depicted in red boxes. The amount of the essential fatty acid precursor, linoleic acid, is about 1/10 the concentration found in soybean oil emulsion, but is still sufficient to prevent essential fatty acid deficiency. In addition, the amounts for the true essential fatty acid, i.e. arachidonic acid, from which linoleic acid is converted, is about 2%, whereas only trace amounts are found in plant-based triglycerides. Here are the fatty acid profiles for 2 mixed oil injectable emulsions that also contain fish oil. Of the oil phase of the emulsion, Lipo Plus or Lipidem contains 10% fish oil, while Lipid contains 15% fish oil by weight. Again, clinically significant amounts of the essential fatty acids are present in these mixed oil products. Of note, arachidonic acid constitutes about 0.5% of the total fatty acid profile from fish oil for both emulsion products. Continuing with this comparison, and although Lipid comprises 50% more fish oil in the oleaginous phase than Lipo Plus, that is 15% versus 10%, more EPA and DHA is present in Lipo Plus, as highlighted in the blue boxes. This is due to final product specifications, which are in compliance with the separate monographs. When the data is normalized to the fish oil fraction for both products, they do meet the minimum requirements for each European Pharmacopoeia monograph, as noted on the right side of this slide. Although the main focus of this talk has been on the composition of injectable emulsions, I would be remiss were I not to mention the factors associated with its safe administration. This schema relates to the metabolic disposal of intravenously supplied lipid emulsions and is relevant to all compositions upon intravenous infusion. If these factors are uncontrolled, they can produce fat overload syndrome with severe metabolic and physiological complications. Clearly, as with any pharmaceutical, the daily dose of lipid injectable emulsion must be carefully determined, particularly during acute illness. What is equally important, however, is the infusion rate, its formulation, and the quality of the emulsion, and how these factors influence the patient's ability to clear lipids from the bloodstream. In the clearance of lipid from plasma, the liver is central to maintaining normal triglyceride levels. And when fat accumulates here, it triggers oxidative stress, inflammation, and immune activation. In the next few slides, I will discuss some of these scenarios and their clinical consequences. consequences. For example, this slide depicts the influence of infusion rate and was based on soybean oil emulsions as it relates to the incidence of adverse events involving vital organs and platelet function. The infusion rate should not exceed 0.11 grams per kilogram per hour, equivalent to, for example, 7.7 grams per hour in a 70 kilogram patient, as recommended. Note that fish oil emulsions did not enter the market until 4 years after this review was published, so this upper infusion rate may not be applicable. Excessively high infusion rates of soybean oil emulsions during discontinuous infusion impairs reticuloendothelial system, or RES, clearance, mainly by phagocytic uptake of fat by the Kupffer cells. These are innate immune cells, and technetium sulfur colloid clearance is a marker of its uptake by Kupffer cells. During a 3-day discontinuous infusion over 10 hours, RES clearance of technetium sulfur colloid was reduced by 50% when measured pre to post-infusion of the soybean oil emulsion. No change in TSC clearance was detected when the same infusion was given continuously over 24 hours at the same daily dose. When first developed more than 60 years ago, soybean oil emulsions contained the same concentration of the emulsifying agent egg phospholipids at 12 grams per liter for both 10 and 20% weight volume formulations. The excess and free phospholipids present in the 10% emulsion form large amounts of micelles as nano-sized liposomes compared to the 20% emulsion, which interfere with the action of lipoprotein lipase and thus impair plasma clearance. Hence, 20% fat emulsions are preferred. Although this is particularly true for infants, there is also evidence of this in adults as well at high infusion rates. When this happens, the role of the RES in clearance of lipids also increases. This stylized plot illustrates the rate of plasma clearance according to the type of emulsion infused over time. Increasing the hydrocarbon chain length of the fatty acid triglycerides prolongs the clearance time. Therefore, medium chain triglycerides consisting mainly of 8 and 10 carbon fatty acids are fastest, followed by plant-based 16 and 18 carbon fatty acid containing long chain triglycerides, and finally 20 and 22 carbon marine-based long chain triglycerides are the slowest. Finally, a spike in the incidence of hypertriglyceridemia was noted by our neonatologists. This study assessed the impact of 2 different soybean oil injectable emulsions on the market, one packaged in conventional glass bottles versus one packaged in plastic containers. We assessed the differences in the globule size distributions in the laboratory and discovered that the lipids packaged in plastic were significantly more coarse than lipids in glass, according to USP limits for the large diameter population of fat globules greater than 5 micrometers, or the PFAT5. The PFAT5 parameter is described in detail in USP Chapter 729 and is intended to limit the concentration of these very large diameter fat globules relative to the mean droplet diameter, which must be less than 500 nanometers or 0.5 microns. We found that infusion of the coarse emulsion was associated with a significant increase in the incidence of hypertriglyceridemia. This enhances the participation of the reticuloendothelial system in its clearance from plasma, that is, uptake by the liver versus its metabolism by lipoprotein lipase. In summary, with respect to intestinal failure associated liver disease from parenteral nutrition, soybean oil as a fat source carries the highest risk. This likely results from a combination of pro-inflammatory substances unique to soybean oil as listed in this slide: omega-6 fatty acids, saturated long-chain fatty acids, particularly palmitic acid, oxidation products, and phytosterols. In conclusion, of the available sources of injectable triglycerides and associated phytosterol levels, soybean oil is the main culprit. But as mentioned, there are other contributing factors or critical quality attributes that may be equally relevant. Second, efficient methods for extraction of phytosterols from plant-based oils are not feasible at this time. Third, reducing or eliminating soybean oil from the composition does not necessarily incur clinically significant risks. Moreover, mixed oil injectable emulsions currently dominate the clinical market. Finally, if the agency desires specific pharmacopoeia limits for the relevant triglyceride oils employed in lipid injectable emulsions, the task should be given to the USP committee responsible for monograph modernization. Thank you for your attention.
Thank you, Dr. Driscoll. Our remaining speakers come from the two of the manufacturers of the intravenous lipid emulsions. First up is Dr. Martin Westpal, who is Chief Medical Officer and Executive Vice President for Medical, Clinical, and Regulatory Affairs Business Unit and Nutrition at Fresenius Kabi in Germany. He's also a professor at the Department of Anesthesiology, Intensive Care and Pain Medicine at the University Hospital of Münster in Germany.
Dear ladies and gentlemen, warm welcome also from my side. My name is Martin Westphal, and I would like to thank the FDA very much for the opportunity to present an industry perspective during today's scientific workshop. I will briefly address the pathogenesis of PNALD and IFALD, including risk factors, and then present non-clinical and clinical data relevant to the topic. Thereafter, I will talk about methods that could theoretically be applied to reduce natural content of phytosterols in vegetable oils and discuss also the respective implications. Finally, I will share some important information on clinical trial feasibility to demonstrate a potential safety benefit of phytosterol reduction in lipid emulsions. This slide illustrates the pathogenesis of PNALD and IFALD. The risk factors can be attributed to two major origins, namely patient and nutrition-dependent causes. Without going into the details of the individual risk factors, one can easily see that the development of PNALD and IFALD is multifactorial. This in turn implies that risk mitigation of this disease needs to consider also both patient and nutrition-dependent causes. In this context, it is important to note that non-clinical preterm piglet models only focused on the effects of oil emulsion components. The aim of this non-clinical study was to determine if altering the phytosterol concentration of an intralipid-based soybean oil emulsion alters the onset and severity of direct bilirubin serum levels in parenterally fed preterm piglets. The first group of animals received a soybean oil emulsion which contained 75% less phytosterols compared to the second group that received a soybean oil emulsion with normal phytosterol content. The third group was treated with a 39% phytosterol-enriched soybean oil emulsion. The figure on the right side shows a phytosterol dose and time-dependent increase in direct bilirubin serum concentrations in all groups. Interestingly, the direct bilirubin levels in group 1 on day 21 were comparable to the concentrations of group 2 on day 14. As shown earlier, phytosterols are not the only factor influencing the development of PNALD. In this context, it is important that the phytosterol effect may be counteracted by changing the oil type of the lipid emulsion. In this study, direct bilirubin serum levels were followed for 14 days in preterm piglets fed with total parenteral nutrition that contained either fish or soybean oil-based lipid emulsions. While group 1 received a fish oil containing lipid emulsion, group 2 was treated with a phytosterol enriched fish oil emulsion that contained equivalent levels of phytosterols administered in group 3 by a soybean oil emulsion. As shown in the figure on the right-hand side, there was no increase in direct bilirubin serum levels in piglets treated with phytosterol enriched fish oil emulsion as seen with soybean oil emulsion. Thus, phytosterols do not appear to be the main contributor to cholestasis. Let's continue with the clinical setting. Notably, there are only limited data available on the impact of blood phytosterol levels on PNALD incidence. Among the 6 available publications, there is only one randomized controlled trial which did not show an association between plasma phytosterol levels and cholestasis. Fresenius Kabi also conducted a clinical study to compare safety and efficacy, including risk of PNALD of SMOF-lipid versus intralipid in hospitalized preterm and term neonates and infants. All of them required PN for at least 28 days. This was a phase 3 randomized double-blind controlled multicenter study in 14 academic institutions across the United States of America from December 2015 to March 2020. While 83 patients received SMOF-lipid, 78 were treated with intralipid. In this context, it is important to know that SMOF-lipid contains approximately 60% less phytosterols than intralipid. Relevant endpoints of this study include the number of patients in each treatment group with conjugated bilirubin levels of more than 2 mg per deciliter during the first 28 days of the study treatment period, confirmed by a second sample collected 7 days after the first sample, as well as plasma phytosterol levels. Overall, there were only a few PNALD cases with an incidence of 3.1% over the first 28 days on PN and 6.8% for patients that received PN for up to 84 days. While there were numerically less patients with PNALD in the SMOF-lipid group, there was no significant difference among groups despite the 60% difference in phytosterol content. The squares in the graph represent the phytosterol levels in the 3 study periods, which are from left to right baseline, the end of first 28 days, and the end of 84 days. SMOF-lipid results are shown in blue and intralipid data in yellow. Whereas phytosterol levels remain stable in the range of around 40 to 80 microgram per milliliter in the SMOF-lipid group over time, there was a marked increase in the intralipid group from approximately 80 at baseline to 280 microgram per milliliter at the end of the 84-day study period. The dots represent the individual PNALD cases and the respective phytosterol levels. Whereas more patients suffered from PNALD in the intralipid group as compared to patients receiving SMOF-lipid, a similar amount of PNALD cases was noticed in each period after baseline. Since there was no correlation between phytosterol levels and the incidence of PNALD, these results strengthen the assumption that phytosterols are not the main cause of PNALD. This slide summarizes key aspects relevant to phytosterol chemistry. We know that vegetable oils are the richest natural sources of phytosterols and that more than 200 different types of phytosterols have been identified. Among them, beta, Phytosterol, Campisterol, and Stigmasterol are the most abundant compounds. Soybean and olive oil contain 60 to 85% free and 15 to 40% conjugated forms, for example, esterol esters. Free phytosterols may be partially removed from vegetable oils during refining or after, using additional unit operations. Literature suggests that reduction of conjugated forms is technically feasible, although major reduction requires methods that impair the quality of the refined oil. Therefore, the overall negative impact of the phytosterol reduced oil must be assessed before implementation. Vegetable oil production involves several steps that are necessary to render it suitable for human consumption. The crude oil resulting from the extraction process must therefore undergo additional treatments, collectively referred to as refining, to remove undesired ingredients. The major steps of oil refining and the main components removed during the respective process are shown on this slide. Phytosterols may be partially removed in the three of the unit operations used in the oil refining process, namely neutralization, bleaching, and deodorization. The overall phytosterol reduction varies and depends on the processing conditions used in the various stages. However, it needs to be considered that neutralization requires a chemical step in the refining process. During the bleaching step, the oil is heated and treated with acid-activated bleaching earth, which is linked to the formation of nonpolar sterols with pro-oxidative activity. Deodorization of oils is usually based on steam distillation at elevated temperature under reduced pressure. Consequently, the sterile ester fraction increases and thermal oxidation products may be generated. Long deodorization times and/or too high temperatures can have a dramatic effect on the oil quality, not only because of the chemical changes, but also due to the distillation of a significant part of the natural tocopherols. In this context, it needs to be considered that removal of tocopherols decreases the stability of the refined oil. In addition, removal of tocopherol and stressing processing conditions favor the generation of thermal oxidation products. A key question is now, what are the overall medical implications of phytosterol reduction? We know that phytosterol oxidation products may undergo complex chemical reactions during the refining process, but the generation of degradation products and the underlying mechanisms are still not fully understood. However, thermal oxidation products of phytosterols have been associated with health hazards and safety concerns. Additional purification operations could be adapted to further reduce phytosterol concentration in the refined oil. But the use of multi-stage processes at high temperature can induce formation of trans isomers, triglyceride dimers, or other degradation products that are also linked to safety concerns. The bottom line is that clinical implications in terms of benefit-risk assessment of a partial reduction in phytosterol content remain unclear. When considering designing a clinical trial to demonstrate a safety benefit of a phytosterol-reduced lipid emulsion, we should remember that the incidence and observed effect size in Fresenius Kabi's previously mentioned randomized clinical trial were very small. Using this effect size for sample size calculation reveals that such a clinical trial would require to enroll almost 4,000 patients, which is not feasible in a meaningful time. Let me summarize and conclude. As the pathogenesis of PNALD and IFALD is multifactorial, risk mitigation should consider both patient and nutrition-dependent causes. Non-clinical studies showed that even a marked reduction in phytosterols did not achieve a no-effect level. Thus, phytosterols are obviously not the main contributor to elevated direct bilirubin levels. Notably, published clinical trials did not reveal a clear association between phytosterols levels in different lipid emulsions and the incidence of PNALD. In Fresenius Kabi's randomized control trial, fewer PNALD cases were observed in the lipid group. However, there was no statistically significant difference among groups despite a 60% difference in phytosterol content. Phytosterols are already reduced in vegetable oils during the refining process. Further reduction is theoretically possible, but practically linked to undesired effects. Clinical implications regarding the benefit-risk of phytosterol reduction in lipid emulsions are uncertain, and a clinical trial to evaluate a safety benefit is not feasible because of the very large sample size. I thank you very much for your attention and look forward to receiving your questions.
Thank you, Dr. Westpal. We want to clarify a point that Dr. Westpal made. The clinical trial described by Fresenius Kabi was originally powered for a statistical analysis. But because there were so many fewer PNAC cases than expected in the interim analysis, the design was changed to be a descriptive study. So when Fresenius Kabi states that there was no statistically significant difference in PNAC between phospholipid and intralipid, the study was not powered to be able to make a conclusion about statistical significance. Our final speaker this afternoon is Mr. Baptiste Baudet, who is a chemical engineer from National Institute of Applied Sciences and works at Baxter's Clinical Nutrition Business Unit, Research and Development Department as a technical lead, accountable for technical strategy and development.
Good afternoon, good evening, everyone. We are here today to discuss about the role of phytosterols in parenteral nutrition-associated liver disease, also known as PNALD, and intestinal failure-associated liver disease, IFALD. First of all, I would like to thank the FDA and the members of the workshop today for giving Baxter the opportunity to share insights on the role of phytosterols in PNALD IFALD. My name is Baptiste Baudet. I am a chemical engineer working as technical lead in the R&D department of Baxter's Clinical Nutrition Business Unit. Today, I would like to present three main topics, starting with general information on phytosterols, before moving to the composition and role of specific compounds in US intravenous lipid emulsions, abbreviated as ILE. Finally, I will present you the scientific challenges and risks associated to producing a phytosterol-reduced ILE. I would like to start this presentation with a general overview on phytosterols. The first key takeaway of today's presentation is that phytosterols come from plants and can be found in a wide variety of food products. More importantly, phytosterols are natural components of the diet. Their content depends largely on the type of foods ingested, knowing that most of phytosterols originate from vegetables, fruits, nuts and seeds, legumes, and oils. Many foods are also fortified in phytosterols. This is notably the case for bread, cheese, and biscuits. As you can see in this table from the Yasilo-Siegel paper of 2017, every diet from non-vegetarian to fully vegan contains a consequential level of phytosterols. Finally, an important point to mention is that phytosterols have been associated to numerous health benefits, such as lowering LDL cholesterol and coronary heart disease, as well as a lowering of rates of certain cancers. In the next slide, I would like to compare the phytosterol intake in adults and children from both the diet and intravenous lipid emulsions. It should be noted that several expert associations, such as the National Cholesterol Education Program, the National Lipid Association, and the European Atherosclerosis Society, consider a daily intake of 2 to 3 grams of phytosterols to be a heart-healthy diet, and having the potential to reduce LDL cholesterol levels by up to 15%. Let's take a concrete example, in adults first. What you can see is that the dietary absorption of phytosterols exceeds the phytosterols intake from IV lipid emulsion. This has been calculated considering a 2,000 to 4,000 milligram per day dietary phytosterol intake and an absorption of 7%, compared to an IV lipid intake in adults of 1.3 gram per kg per day of commercially available ILE and its respective phytosterol content of 180 to 340 microgram per ml. Now, if we take neonates and children, the Cleveland Clinic cited an acceptable daily intake of phytosterols for children to be 130 milligram per kg per day. If we take the example of infant formula, you will notice that they contain a significant amount of phytosterol, as you can see in this table summarizing a few studies that have measured the phytosterol content of certain formulas. If you look at the IV intake of phytosterol from ILE for neonates, you will notice that it is in the range of phytosterol absorbed from infant formula. Moreover, the phytosterol intake from ILE stays lower than the acceptable daily intake in children cited by the Cleveland Clinic, which is around 9.1 milligram per kg per day, considering an absorption of 7%. Today, the 3 most common phytosterols in lipid emulsions are beta-cytosterol, campesterol, and stigmasterol. Other phytosterols are also present. However, their amount is usually significantly lower compared to these 3. In the table on the left, adapted from a study performed by Yang in 2019, you can see an example of levels of these 3 phytosterols in some widely used oils, notably in olive and soybean oil, with olive oil being in the lower range in terms of phytosterol content. Now, because phytosterols are natural components coming from plants, their amount is not constant across plants and across the years. Several factors impact the phytosterol content in plants, notably the plant variety, the soil composition, and the climate. Based on a study from Kitchik done in 2016 on 43 olive species, the total sterol level among species could vary as much as 3-fold in content. Due to this known variability observed in oils used to manufacture ILEs, phytosterol content are controlled and limits have been established in ILE for the major individual phytosterols. Let's move to the next topic of today's presentation about the composition and role of specific compounds in ILEs. Based on the publication from Shu in 2012, you can see the phytosterol levels present in 3 ILEs available on the US market: a pure soybean oil-based ILE, a mixed oils ILE containing soybean oil, medium-chain triglycerides, olive oil, and fish oil, and finally, a predominantly olive oil-based ILE containing olive oil and soybean oil. As you can see, the phytosterol level is higher in pure soybean oil-based ILE compared to new generation ILE made of 4 mixed oils or olive oil mainly. These new generation ILE have phytosterol levels that are roughly 30 to 50% lower than pure soybean oil ILEs. As mentioned in the previous slide, you can observe again the 3 major phytosterols, with beta-cytosterol being the most predominant one. Among these major phytosterols, it is also striking to see an almost 2-fold difference between the mixed oils emulsion and the olive-based emulsion for both campisterol and stigmasterol. These individual differences might be important, as existing literature varies on the importance of each phytosterol. Additionally, the essential fatty acids are not the only beneficial compounds of ILE, and other essential compounds are also present in various amounts. For example, the tocopherol levels of these three ILE are quite different, with mixed oils lipid emulsions being highest in total tocopherols. The cholesterol amount in mixed oils ILE is also significantly higher due to the presence of fish oil. However, as you can see, the squalene level of olive oil-based emulsions is consequently higher than in the two other emulsions. We will review in the next slide the benefits linked to these essential compounds. ILE are made of different types of unsaturated fatty acids in the form of triglycerides. The omega-6 and omega-3 PUFAs are the preferential targets for oxidation due to their multiple double bonds, exposing them to free radical attack and peroxidation. MUFAs, like the oleic acid, majorly present in olive oil-based emulsions, are less prone than PUFAs to lipid peroxidation due to their single double bond. Oxidation occurring in the oils and ILE is a chain reaction, as the hydroperoxides formed from the oxidation of PUFAs can themselves act as oxidizer for other PUFAs. On the right, you can see the full oxidation pathway of PUFAs, with notably the formation of conjugated dienes and lipid hydroperoxides. Consequently, oxidation in ILE should be avoided, as the hydroperoxides formed lead to the formation of free radicals capable of oxidizing the tissue component and thus representing a risk for the patient. For this reason, oils contain natural antioxidants that are key to protect them from oxidation. The main antioxidants found in ILE are sterols, notably vitamin E, phytosterols themselves, and cholesterol, as well as squalene, mentioned in the previous slide. Squalene is a precursor of lanosterol, which itself is a precursor for cholesterol. Thanks to its multiple double bonds, 6 in total, squalene acts as a strong antioxidant and is therefore a key component of ILEs. Now, the key takeaway that should be kept in mind for the next slides is that removing any one of these compounds would influence the antioxidant properties of ILEs and therefore impact the quality of the oils. On this slide, you can see two concrete examples of the importance of squalene in ILE. The first study, on the left, from Somaidal, has studied the evolution of the peroxide value in olive oil at 40 degrees Celsius in presence of different concentrations of squalene. This study has shown that the peroxide value increased slower with increased levels of squalene in the olive oil. The second example I would like to present is a study from Conforti, who studied the inhibitory activity of squalene on lipid peroxidation. This is summarized in the graph on the right, where they have observed an increased inhibition of lipid peroxidation in liposomes with an increasing squalene concentration, reaching around 90% inhibition with a concentration of squalene of 0.1 milligram per ml. Now, as a reminder, squalene concentrations in new generation ILEs range from 0.05 milligram per ml in mixed oils ILEs to almost 0.4 milligram per ml in olive oil-based ILEs. These studies emphasize the importance of squalene at levels currently present in the intravenous lipid emulsions and its major role in preventing oxidation. Now, phytosterol also have their own importance in ILEs. Based on a study from Mortimer in 1995, it has been observed that phytosterols play a similar role than cholesterol in the clearance of lipid particles in the bloodstream. As you can see on the graph, cholesterol is represented by the dotted line, and the various phytosterols have a very similar clearance rate as cholesterol. Therefore, removing phytosterols might lead to clearance issues for patients, notably in infants. I would now like to move to our last topic for today, focusing on the production of an ILE with a reduced phytosterol content. Based on recent discussions around phytosterols, Their removal from ILEs has been suggested. It should be kept in mind that removing phytosterols from the oils and ultimately from the ILE is not simple and implies adding a process step during their manufacturing. These additional process steps might impact the characteristic of the oils and eventually the quality of the final ILE. If these new process steps are utilized, some of the risks to be considered are: first, the removal of essential compounds acting as antioxidants, for example, squalene and tocopherols that we just talked about. Second, the potential oxidation of the oils, notably through the generation of hydroperoxides and/or conjugated dienes, as well as potential other risks, such as an impact on the clearance of lipid particles in the bloodstream. To conclude, I would like to highlight the key takeaways of today's presentation. As reviewed today, phytosterols are recommended components of a normal diet and can be found in many foods, including infant formula. They have been associated to numerous health benefits, such as lowering LDL cholesterol and the rates of certain cancers. As there are natural components that originate from plants, their level in foods, and notably in oils, is very variable. Therefore, they are controlled in currently marketed ILEs. Also note that new generation ILE has phytosterol levels at least 30% lower than the current standard of care made of pure soybean oil. As discussed previously, the unsaturated fatty acids present in the oils are sensitive to oxidation due to their double bonds and can lead to the formation of free radicals, which are capable of oxidizing the tissue components. This highlights the importance of avoiding oxidation in ILEs. Now, apart from phytosterols, which are natural antioxidants, oils used to manufacture ILEs contain other essential components that also act as antioxidants. This notably includes tocopherols, squalene, and cholesterol. Finally, it should be kept in mind that the phytosterol depletion process is not simple and may impact the characteristics of the oils and therefore the quality of the ILE. This process might be associated to new risks, such as the oxidation of the oils, the removal of essential compounds acting as natural antioxidants, and the impact on clearance of lipid particles in the bloodstream. Consequently, the removal of phytosterols imparts additional risks to the ILE that may outweigh the potential benefits of lower phytosterol levels. I would like to thank the FDA once again for providing the opportunity to Baxter to share our thoughts, and thank you all for your attention.
Thank you, Mr. Bidet. Please take the next 10 minutes for a stretch break, and then we will come back for the panel discussion. Thanks to all the presenters for excellent talks. We welcome all the panelists and look forward to a robust discussion. Two panelists who did not give talks this morning include Dr. Valeria Koran and Dr. Muralidhar Premakumar. I would like to introduce Dr. Koran. She is an associate professor of pediatrics at the Feinberg School of Medicine and the medical director of intestinal rehabilitation and transplantation at the Anne and Robert H. Lurie Children's Hospital of Chicago. Dr. Koran, would you please introduce yourself and take a few minutes to share your initial thoughts, and also please state if you have any disclosures. Dr. Koran.
Thank you so much, Dr. Mehta. As you stated, I'm from Chicago, Lurie Children's Hospital here. My disclosures are such: I've served on the Speakers Bureau for Abbott, for Nutricia, and for also Takeda. Listening to this excellent workshop today, there's so many great talks talking about the preclinical and the clinical aspects of phytosterols and their potential role in intestinal failure-associated liver disease. All the speakers have been very detailed, and it's very interesting when you see how much data there is, but we really don't have a lot of randomized controlled trials in pediatrics regarding some of this data, despite the fact that we do have a decent number of children, but I think that, it becomes hard in pediatrics, especially when drugs have been already approved or being used off label. One of the prior comments that was made was about education and really trying to educate people who take care of these patients about intestinal failure, because there are still instances where children perhaps their care could be optimized. And Dr. Sam Kokoschis, who was one of the prior speakers, actually talked about in 2004 with the white paper about improving education for these patients, or for the people who are taking care of these children. Dr. Iyer, who's one of the panelists, actually started a program called Pediatric Lift Echo, so learn intestinal failure in pediatrics. And so that's an organization a meeting that occurs twice a month. We discuss different cases from people throughout the world to try and really improve our educational knowledge about intestinal failure, associated liver disease, and the management of those patients. And I think education is so important, including workshops like this, to be able to improve the care and optimize the care of these patients. Thank you.
Thank you. Dr. Prem Kumar is an associate professor in pediatrics and neonatology at Baylor College of Medicine and Texas Children's Hospital. He's also the co-director of the Intestinal Rehabilitation Program. Dr. Prem Kumar, would you please introduce yourself and take a few minutes to share your initial thoughts?
Good afternoon, everyone. Thank you for inviting me to this symposium, and it's an honor. And I'm really so pleased to be part of this elite group. I'm Murali Prem Kumar. I'm an associate professor here at Baylor College of Medicine and Texas Children's Hospital. And as Dr. Mehta mentioned, I'm also the co-director for the Intestinal Rehabilitation Program. And I'm also the associate program director for the neonatal perinatal medicine fellowship program. This was a fascinating day of wonderful talks with the world experts in this field. And we discussed the deficiencies in the definitions of intestinal failure and intestinal failure associated liver disease. We discussed how quickly this field has been changing over the last 15, 20 years. And that has made it very difficult to capture the prevalence of the problem we are trying to solve here. And we also discussed about the difficulties in the biochemical assessment and the clinical evaluations of these babies with of children and adults with intestinal failure associated liver disease, and the difficulties in performing studies, particularly when the effects are modest, how difficult it is to perform these randomized controlled trials to get the gold standard evidences. And we are also trying to decide what are the outcomes which we have to look for as we decrease the mortality and improve the outcomes in these children. And we discussed the treatment and also some of the future directions. There were many areas of minor disagreements, but I think one common thread of agreement, which I thought several speakers made their points, was while phytosterol is probably or undoubtedly one of the contributors, there are other factors other than the phytosterol excesses. And I think this is a great symposium to start thinking outside the box. One final point I would like to make was, I think it was aptly made by the European here in the panel, is about the prevention. We as Americans talk about the treatment and disability limitation, but we also need to look at various modes of prevention of this intestinal failure associated liver disease. Thank you once again for the opportunity.
Thank you. I'll go in the following order. Dr. Burrin, if you could respond, followed by Dr. Iyer, and then Dr. Driscoll, and then Dr. Koran, and Dr. Prem Kumar. If you have a comment, you can raise your hand. The opening question to the whole group is, does the non-clinical and clinical evidence support phytosterols as dose-related hepatotoxin? Dr. Burrin?
Yeah, so I really appreciate the opportunity to be here with this group at this workshop today. It's been really illuminating. I've learned a lot, actually. So in answer to the question, I feel like there is fairly solid evidence that there is a correlation between accumulation of phytosterols or phytosterolemia, however you want to term it, and the accumulation or increase in markers of liver injury, bilirubin, bile acids, GGT. A number of those have been discussed today. So I do think there is a correlation there. I think what is missing from a lot of the literature, although it is in some of the studies from ours and the clinical studies from the Finnish group, is evidence of actual histopathologic injury and whether that injury is persistent and irreversible. So I think that is something where I think the jury is out, but I think there's a clear correlation between accumulation of phytosterols and markers of cholestasis. And I think now, from a lot of the work that's been done through our work in pigs and the work in mouse models with Ron Sokol's group, really show us some pretty good mechanistic models to explain how this is occurring. So I think going forward now, the question is whether there is an actual definite dose response here, because I think the problem with this condition is it's not only a function of the concentration of phytosterols, it's a function of load and duration of exposure. And so for infants that might be exposed to phytosterols for short periods of time, either with intralipid or with small lipid or multi-component emulsions, this may not be as much of a concern as those infants who are going to be exposed to higher phytosterol containing emulsions like intralipid or soybean emulsions for long periods of time. And then, so I think it's been good today. I also appreciate the comments from Dr. Westpal about the challenges that we have. So I'm looking forward to discussing that.
Dr. Iyer?
Thank you. I want to echo Dr. Burrin's comments. This has been a fantastic workshop, some great lectures. I've been passionate about this for many years and I'm constantly amazed at how much I don't know. And Dr. Burrin is a very hard act to follow. Both a fantastic lecture and very insightful comments. I will say there are a few things that are clear. One, the problem is much better than it was 20 years ago. Dr. Mercer, who was on the panel this morning, now runs the program that I started about 23 years ago. And the phenotype of patients we're seeing with advanced PN-associated liver disease has completely changed. That's a rare patient that you cannot reverse or prevent liver disease. That said, the problem has not gone away. I will submit to you that roughly 40-45%, and that number has not changed, roughly 40-45% of patients getting intestinal transplants in the US are requiring simultaneous liver replacement. That in itself may not seem like a big problem, and you think that there are falling numbers of intestinal transplants. I think last year, 125, 150 intestinal transplants. So we might still be talking about a small number of patients, but try telling that to the family members of the patients who die on the transplant waiting list, unable to get a liver-containing organ. Last year, I had 4 young adults on my transplant list awaiting combined liver intestine transplants. 3 of them died on the waiting list before getting access to transplant, and the 4th had an aborted transplant because of advanced liver disease and inability to safely complete a multi-organ transplant. So while the problem is attenuated, by no means has it gone away, and we'd be burying our head in the sand to say the problem doesn't exist. We've got all these lipid emulsions, we've become smarter, some of us have become gray-haired, we know how to handle this, and that there is a lot of truth to that. Equally, despite spending 4 years of my life trying to prove the phytosterol story, there is no question that there are other factors. Sepsis is such an important factor. We've heard more than once today about a second hit. Dr. Burrin's work is really fantastic. We also know that really the patient you can give any amount of intralipid for 4 weeks and not produce IFALD or PNALD. The problem patient is the premature neonate who has no bowel and you cannot feed, or the patient who needs long-term PN. I have a 2-kilo baby in the NICU with duodenum, anastomosis rectum, and a bilirubin of 10. Tell me that this is not a problem. Tell me that this is a problem that lends itself to an easy solution, and we might cut short this workshop. I'll stop there.
We fully acknowledge that this is certainly a problem. What we're trying to quantify here is what is the magnitude of the problem? Is it the numbers are getting smaller? So which is kind of good to know. But moving on to, go ahead, Doctor.
May I just follow with one quick comment to your numbers issue?
Sure, please.
I served 2 terms on the pediatric committee of UNOS. I served in the policy oversight committee. One of the problems we deal with, and Dr. Corin alluded to this, very, very difficult to do randomized controlled trials in an ultra-rare disease like intestinal failure, especially so when you're talking of an uncommon problem in an ultra-rare disease. If we wait for randomized controlled trials to prove the evidence that we have in front of us, we will do a disservice to another generation of intestinal failure patients. It may be that we have all the solutions, and what we need to know is how to deploy them or how to use them. But I don't think this is a numbers issue.
Sure, I agree with you. Dr. Driscoll, we'd like to hear your thoughts.
I would concur with the other speakers, and I made the point in my slide. There are many reasons, as I call them, pro-inflammatory substances that are present in the emulsions, depending on the oil composition. So yes, I believe the story of phytosterols is real. I also believe that there's more than a second hit. There are several hits, particularly from a chemicals perspective, not including the predispositions of the patient condition itself. Thank you.
Sure. Thank you very much. Dr. Corin, would you like to go next?
I'll simply echo what the previous speaker said, and also, the fact we're saving younger and smaller infants than that what we were even saving 20 years ago. So yes, to a certain extent, it has decreased. But as Dr. Iyer mentioned, to have an infant that's 2 kilos and has no intestine, we're seeing more and more of that. Those are the kids who are at risk. And so I think, paying attention to the phytosterols, the amount that we're providing them, trying to use some of these alternative lipids are very important as part of their therapy.
Sure. Dr. Prem Kumar?
Yeah, I concur with all the speakers who have said so far, who have stated their opinions so far. And as Dr. Iyer and Dr. Corin and others mentioned, the frequency might be getting less, but the denominator is getting larger. We mentioned about we are extending the thresholds of prematurity to 22 weeks. And as you push the thresholds of prematurity, you see more and more intestinal perforations, you see necrotizing enterocolitis. And as we know, the frequency of these diseases are not only much more common, but are also much more severe in extremely preterm babies. And compared to, say, even a decade ago, we are able to help these babies survive and help these babies survive with patients like Dr. Iyer mentioned, with almost no small intestine at all, or both small intestine and colon. So ultra short bowel syndrome is a fairly recent term, and even we are finding terms to describe those patients who have no short bowel now. What do you call them? So with these patients, this body of patients who need help with their challenges is definitely going to grow. So we need to look closer at these solutions.
Yeah, so that leads me to the next question. Is a phytosterol depleted lipid formulation scientifically feasible? Dr. Burrin, you could take a crack and then.
Yeah, so I'm probably not the best expert on this panel to answer that question. I would probably defer to Dr. Driscoll. But as you saw in the study that I highlighted that was done in collaboration with Fresenius, we were not able to get the phytosterol content completely down to zero using soy emulsions. So So I think that is a challenge, and I think Dr. Westphal and Berto kind of highlighted some of the challenges and pitfalls of even trying to do that. And so I think one of the things that if we really feel like it's important to substantially lower phytosterols, lower and still maintain the important fatty acids that we need to give, for both energy and essential fatty acids. The question is, can we consider, a multifactorial approach where we look at different lipids that have low phytosterols, but also can provide essential fatty acids and important N3 fatty acids. So maybe an optimized blend to achieve this. But I think starting with high phytosterol-based emulsions and trying to, deplete them seems like a real chemical challenge. So I'd defer to Dr. Driscoll on that.
Sure. Dr. Driscoll?
Well, it's from a feasibility standpoint, and as you saw from both of the folks from industry, there are consequences to doing these type of manipulations. There are extra procedures, and in fact, you can produce pro-oxidants. So not only is it difficult, whether it's non-enzymatic distillation or enzymatic lipase, on an industrial scale, there does pose significant problems. On a practicality scale or safety scale, it carries risks. So I don't think we're really close to being able to successfully do this in a way that does not adversely affect the oil and subsequently the emulsion safety profile.
Does anyone else on the panel would like to take a crack at the question?
Dr. Mehta, not to take a crack at the question because I can claim no expertise, and I'll tell you whatever Dr. Driscoll said. But I was intrigued to follow with Dr. Burrin's comments. They echoed something Dr. Martin had said in the morning, and I think it was, to me, an interesting conceptual framework, even though we do it quite often without thinking in those terms. She spoke of a maintenance regimen, and I think she called it a therapeutic medication, and maybe that is the answer. Maybe I could see the defense. This is an opinion. They're not on a panel. They can't yell at me, I guess. But I sensed a defensive posture from the manufacturers, and maybe this is a challenge. They have told us what their challenges are, and maybe the answer is to take a path like Dr. Burrin suggested. We have plenty of lipid emulsions. We have a good understanding of the problem. Maybe clever people need to come together and say, how do we use these in what situations?
That's an interesting approach.
I would also like to make a comment here, and there's seems to be some misconceptions, and I tried to bring out the difference between a precursor and a bioactive. And there's suggestions that if you don't have linoleic acid or alpha-linoleic acid, then they're going to develop essential fatty acid deficiency. Well, they're called precursors for a reason. They have to be converted via elongases, adding 2 carbon units to get to EPA for the 20 carbon compound in terms of ALA, and arachidonic acid, of course, 20. But to get to DHA, it's additional 2 carbon units that have to be attached. And I think that the reason that they are called precursors is because it's that step that's required. It's EPA that has the effect on leukotrienes and prostaglandins. It's DHA that has the effect on pro-resolving molecules. It's not the precursor. So I think by reducing the soybean oil fraction or any or other oils that are high in phytosterols does not compromise the formulation. And let's not forget, when we talk about fish oil, you also have linoleic acid in there, as well as you have arachidonic acid, the true essential fatty acid. And if you go back, if you look, it's about 1 to 4%. I saw somebody put that up in terms of the percentage of calories from linoleic acid. Well, 1 to 4% is a minuscule amount. And again, that's of the precursor, but you already have the arachidonic acid in there. Now, that's only for the marine oil-based formulations. But I think a mixed oil formulation, rather than
all of the efforts that seem to want to do some type of extraction procedure, which will be extremely costly and possibly detrimental to the chemical and safety of the oil, I think a mixed oil preparation is clearly sufficient. And as I said, you saw even in just the pure fish oil, there's over 4% linoleic acid there. At the break, I just did a quick calculation, and 4%, if you had a 50 kilogram adult patient getting 25 kilocalories per kg per day, that would be 1,250 calories. 4% of that, in terms of the amount of soybean oil that you have to give, is about 10 or 11 grams, because half of that soybean oil fraction, at least half, is linoleic acid. So it's a very, very small amount in terms of precursor. And when you do have the actual essential fatty acids there present, those are the bioactive compounds.
Dr. Karan, do you have any comments for that question about the phytosterol depleted compounds?
I'm sorry, did you ask me?
Dr. Karan.
Okay.
No, I don't have anything further to add.
Sure, thank you. Dr. Prem Kumar, do you have any?
Probably I don't know if this might lead into the next question. We tend to think of these associations as linear. That's because that's easy to assume, right? But I don't think it's linear. Probably, I hypothesize, I suppose that this is an S-shaped curve. What I mean by that is probably, I don't know if I'm holding this up.
Yes, we can see that.
So I think, we were probably somewhere in 1990s high on the S-shaped curve, and we've already pushed it down to 2022 now. And now I wonder if we're pushing it down further, we are going to decrease the morbidity any lower. I doubt.
Interesting. That's a very interesting thought. So that does lead us into the third question, which is demonstrating a low phytosterol content adequate to establish the safety of any intralipid emulsion? Or must such a product be demonstrated to have safety advantage in a clinical trial? So I guess you all have answered it partially, or actually to quite a full extent, but if anybody has any further thoughts on that question, otherwise we could certainly move on to the next one. But go ahead, please.
Sorry. So what I was going to say is, we have already dropped it by 50 to 60% with our SMOS lipid compared to the intralipid. And even with the Cochrane reviews, which Dr. Martin quoted, that had at least 6 or 7 studies with around 600 children, head-to-head comparison with SMOS and intralipid, and there was no difference, meaning the clinical evidence, the clinical effect from these interventions have already been, I think we have seen, and they're so modest. These clinical trials might not be powered enough to pick up these modest improvements in clinical outcomes.
Let me ask a question over here. If we use the real world evidence, or could that provide us some answers? Because then you won't have to power the studies in terms of you can randomize all the patients to a phytosterol depleted product.
I was going to make a point, something related to this, because some of these studies which Dr. Iyer and many of the other speakers mentioned was like the Kavi study or the Colomb study and Allardyce study. I think what you're trying to say is also, I think we have reached the limits of these randomized controlled trials, and we have to look at the epidemiological studies now, use big data to get huge numbers, large consortiums get together and try to answer these questions. And I think that's one of the ways forward. I don't know if that kind of answers your question.
Yeah, it does. But just one last time, I'm going to reiterate the question. Would conducting a trial with phytosterol depleted product be even feasible in 2022?
Really, if I may. So
yes,
I'm involved with now multiple trials, not in the context of liver disease, though maybe one is coming, but in the context of intestinal adaptation and GI peptides. And these are incredibly challenging trials to run. I was involved in the pivotal trial that led to Sorona-Zopte being approved in 2000 or 2001. That trial took 10 years to complete in a small number of intestinal failure patients. We've just had the STEPS trial took several years, nearly got shelved before teduglutide was approved. We've just had one longer-acting GLP-2 trial cut short prematurely because of recruitment, of course, compounded by COVID. These complex clinical trials, and frankly, the level of data that the FDA requires in such ultra-rare diseases and the reluctance to accept surrogate endpoints is an absolute obstacle to such trials. When you look at the complexities that Dr. Driscoll spoke to, that we speak of phytosterols as one thing, and we've heard there are multiple phytosterols, but you move the phytosterols, you change other things. I can't even begin to imagine what such a trial would look like. And we heard from Fresenius that a trial might take 4,000 patients. I don't know where that number came from, but I can imagine that complexity of scale. So the short answer is no. I don't think a trial is feasible.
Yeah, I would agree with that. It seems like, and I think Dr. Westpal, showing the RCT data from their study and showing the power that was just alluded. I mean, if it is going to take 4,000 patients to do an adequately powered study, given that the incidence of this condition is now low enough to require that many infants, then it's going to be really hard pressed for any industry company to spend the money to do that. So I think then we're left back with a couple options. Number one, making historical retrospective comparison of SMOF treated cohorts to intralipid treated cohorts, which I think is also confounded by the fact that most of us probably realize, those of you who are clinicians, not myself, that care for these infants is improved, and how these infants are cared for that are receiving lipid emulsions. So it seems like what we're left with at this point, barring the production of any phytosterol depleted emulsions, which seems difficult, is to move forward with some of these lower phytosterol containing emulsions like the multi-component emulsions, and seeing how the incidence of this condition fares going forward. And my question is if you have an infant that's on SMOF, and that seems to be an increasingly popular emulsion that's being used now as a first line of emulsion for preterm infants, especially, which we think is the most critically ill group here to start with, is to see whether, if they're on this for less than a month, whether this is really a problem.
Mm-hmm. Thank you, Dr. Burrin. I would want to loop back to Dr. Iyer, your comment about requiring big trials, requiring thousands of patients. That's not true. FDA does have the ability to be flexible, especially in rare diseases. I mean, we do accept data in very few patients, but the data has to be robust, because at the end of the day, you're going to label. So you need some clinical evidence to support the dosing and administration. So, but we do understand the challenges of conducting a trial in rare diseases. Absolutely. There is a question for Dr. Driscoll. This was asked in the previous session, and I'm going to just repeat the question over here for you, Dr. Driscoll. Does the preparation of parenteral nutrition product that includes lipids have an effect on the product degradation of lipid droplets?
Well, two things I would make. First of all, if you make an all-in-one mixture where you put in the amino acids and dextrose, you've definitely caused massive destabilization, so to speak. But if you, and we published a paper with Dr. Martin, and if you start with an emulsion that's coarse, so it's not unstable, but it's coarse, according to a PFAT-5 standard, versus one that isn't, then we saw that there was a highly significant difference in the incidence of hypertriglyceridemia. And so the ability of lipoprotein lipase to deal with a fat globule that's now no longer submicron, but in the 5 or 10 or even larger microns, it's just not effective. This is going to prompt our reticuloendothelial system, the liver, specifically the Kupffer cells, to assist in its clearance. And then as fat accumulates in the Kupffer cells, that introduces a whole series of complications, not the least of which is pro-inflammatory response, and several other things that can happen. So yeah, that to me, that's the particle size, but there's a USP standard for that. So now all emulsions in the United States should be in compliance with that standard. You mentioned about, just to go back on your original question, is okay, if you want to do a phytosterol study, and I know how long it might take, but you deplete it. What about the other factors? What about the oxidation products that are present there? Those are pro-inflammatory as well. The monograph modernization that's been ongoing has, it's continuing, but I believe that each oil that's going to be used in an injectable emulsion should have its own oil monograph and should have its own standards as to what should be the acceptable levels of these oxidation products. And it will vary because you're going to have the monounsaturates, such as the olive oil containing, all the way up to the super polyunsaturates, including the 5 and 6 double bonds of fish oil. So you might be able to get down depleted, and you have no difference in the results because there's other factors involved that are causing the problem. So I think not only that's another thing that has to be considered if you were to design such a study. We're not ready for that because I still think there's a lot more work to be done, particularly at the USP level, in terms of the ingredients and the monograph of the things that go into this. There's formulation issues, and that's an industry thing. But there are certainly things that can be done now to improve the quality of these products, particularly the raw material.
Thank you, Dr. Driscoll. That was very helpful to hear your response. That is, and also very interesting. I will skip the 4th question because...
Dr. Mehta, sorry.
Yes.
I had one quick comment
Please.
before we...
Yes.
It was for the previous discussion. I'll make it very quickly. I think Dr. Iyer mentioned this ultra-rare disease. What makes it much more difficult? It's very heterogeneous. One patient is different from the other. One does not have duodenum, the other one does not have colon. So I think that makes it very difficult to compare outcomes. And I challenge, I think these numbers are huge because the outcomes are rare. Why don't we go after outcomes that are more meaningful and more common? And probably then we can definitely do a randomized controlled trial with smaller numbers.
Could you repeat that again, the last sentence? I think I missed it. You said.
I think if we wanted to look at the conjugated bilirubin of more than 2 milligrams per deciliter, the care is already so good that there are very few patients who get cholestasis. That's the example with the study Dr. Westpal mentioned. Mm-hmm. And even going back to Dr. Nehra's study comparing omega-1 to intralipid, both at 1 gram per kg per day, the study was abandoned because the incidence of cholestasis was so low. So probably we should go after outcomes which make randomized control trial feasible, but they also have to be meaningful.
I fully agree with you. I mean, you bring up a very good point that the trial should be driven by outcomes, and also you should be able to interpret at the end of the day what the drug is doing or what the or the TPN is doing. So fully agree with that. And that's a challenge with rare diseases. We often face this at the FDA. Most rare diseases are so heterogeneous. The phenotypic presentation is in a rare disease, if it's so heterogeneous, it's very difficult to even think what endpoints you want to use for that population. So yes, I echo the challenges that you have laid out. Moving on to the next question, are there other modifiable risk factors for PNALD or IFALD besides phytosterol exposure? I think we have come to a full circle. We have heard from everyone, but if you have any thoughts, share with us. Otherwise, the next question is, what interventions would you recommend to minimize the risk of PNALD or IFALD?
I think Dr. Iyer wants to make a comment.
Ruby,
Please go ahead.
Two comments. I mean, maybe these two questions are the most important of the workshop. So if we visit them and revisit them, that's not a bad thing. But to Dr. Premkumar's earlier comment about looking at big data, one of the challenges in moving away from smaller studies or RCTs and looking at big data is the fact that we don't have uniformity of terminology until we really don't have an ICD code for IFALD, but if the tea leaves are to be believed, ICD-11 will for the first time have an ICD code for intestinal failure. So it'll be another 5 years, I suspect, before we have an ICD code for IFALD. And then, so when you look at large data sets, for example, third-party claims databases or Medicare data or HRQ data, it's very difficult to track down these patients and to be certain that you're comparing like with like and looking at meaningful outcomes. So that's the challenge. But you asked earlier about real-world evidence. NICE in the UK has made, I think, a significant pivot to saying we're going to evaluate real-world evidence as important, and maybe the FDA can perhaps, this may be one good product that comes out of this, that either there's a white paper or even a follow-up workshop to say what is the real-world evidence in A, B, and C categories that we can look at and then decide what is the next study we need, instead of just saying we need one more RCT looking at the next new epidemiology.
So, Dr. Iyer, this is a great segue for me to say that we do have a guidance. Actually, we do have two guidances on real-world evidence, and you may want to take a look at those. FDA is looking into this, but the characterization of the natural history should be done adequately and appropriately, so that when you come in with the data, that it's meaningful to compare. So those are the elements you may want to consider. But we'll be happy to share those guidances with you. Dr. Kauran, did you want to say?
No, I think we were talking about the modifiable risk factors, and one of the things we haven't really talked about it significantly is actually feeding the infants and trying to feed these children as soon as possible. Now, sometimes there may be surgical complications where you're not able to feed these children early. And I think sometimes we concentrate a lot worrying about the intestinal failure, liver disease. We also need to really, when at all possible, try to advance their enteral feedings as much as tolerated. Because if you can advance their enterohepatic circulation, increase the amount of feeding, promote adaptation, the ultimate goal is hopefully you can reduce the risk of liver disease, and hopefully at some point, depending on the child's status, be able to actually taper them from their TPNs. I think talking about enteral advancement is also very, very important when at all possible.
Dr. Mehta, I think those were really great comments. I think we have to talk more about these other strategies to address this issue. Because, I think we as a nation, more so than any other nation, we are always into disability limitation and rehabilitation. We don't do health promotion, specific protection, or early diagnosis and treatment, meaning we don't spend much time on prevention, and we are also always about treating. And I think there are several strategies. Dr. Kauran mentioned about prevention of this problem by giving more attention to feeding strategies. And I also have, Dr. Iyer here. So we also should look at the surgical strategies to optimize outcome. How can we improve this? I think one of the things which mentioned was, I cannot remember who it was, I think it was Dr. Buchman or Dr. Kokosius who mentioned that, is there room to reanastomose sooner? We are practicing mucus fistula refeeding, where we incorporate the distal component, distal unused ball, so that when we reconnect, we might have better outcomes. How can we salvage greater lengths of intestines? For example, with our babies with neck totalis, previously we would say that there was nothing, the whole gut would be dead. So one look at it, and then we no viable outcomes, and those babies would die. But now with second look surgeries, we give some time, go back in a day or two, inflammation has subsided. Probably that allows us to salvage more wall. So I'd like to hear from Dr. Iyer what surgical advancements have been leading to these salvaging the intestinal length and improving the outcomes.
Murli, I think there's a surgeon trying to get out of you. But you're right. I think one of the things we've learned alongside improvements in understanding of lipid, I think that's just one piece really of what has been almost a 2-3 decade improvement in intestinal failure care overall. I think if there are fewer children coming to intestine transplant, it is because overall intestinal rehabilitation as we know it, medical, surgical, nutritional, has gotten better. That is true for pediatrics, sadly not true for adults. Adult care is still very, very poor. I think NASPGHAN has done a good job of making recommendations on when such kids should go to intestinal rehab programs of excellence fairly quickly, 6 weeks of PN, 8 weeks of PN. We've said here more than once that 4 weeks of PN, you can give a child any amount of lipid for 4 weeks, nothing will happen, may get fat in a short time, but nothing else is going to happen. It's the longer term care that is a problem, and that requires, as we've also said, multimodality treatment to prevent what is a multifactorial problem.
Excellent.
By the way, sorry to belabor this, I just remembered a comment that Alan Buckman made in the morning. We did a survey, so the paper he quoted was our study. We did a survey of 100 US gastroenterologists, roughly 50 of them who claimed they were experts on home PN and intestinal failure. Unfortunately, in a validated knowledge test of intestinal failure, not one scored at the level of experts. So it's distressing. We are paying the price in the US for decentralized complex care.
It seems like there's a lot of work slated out for all of you in terms of coming up to a terminology, coming up to disease progression and identifying, minimizing risk factors and applying it across the whole US, having same standardized procedures. There's a lot of work that needs to be done, for sure. Go ahead.
I was going to add a comment to Dr. Karan's comment about feeding. And I think, feeding is important. Just being able to feed sufficient amounts enterally to reestablish bile flow, you would think practically is going to have an important effect on limiting accumulation of phytosterols. So I think that when I think about that, I think feeding and then also sepsis, inflammation, other things that are more closely monitored on the clinical level. But as I think about this problem, if we accept the fact that we consider phytosterols to be a risk factor, we really don't have good tools right now to measure this quickly and readily in the clinical setting, short of doing LCMS. So that might be something that might be a useful tool down the line, is to be able to develop, an assay for phytosterol, for total phytosterol levels that can be measured. And then we can begin to start monitoring this, on a more widespread basis in populations of babies and infants that are getting lipid emulsions.
Sounds good.
Dr. Burrin, I have a question for you. We were talking about you have an animal model of short gut and intestinal failure-associated liver disease, and we have looked at nutritional strategies. Does your model have the capabilities to look at some of the surgical techniques to see how we can improve the outcomes as well?
Certainly. Just need to find the right surgeon. Fly Dr. Iyer down here, but
I can fly down, Dr. Burrin.
Okay. Sounds good.
I think, as we speak of modifiable factors, all of us on this panel who look after patients, and Dr. Burrin with his piglets, you could I think you can easily conceive of the patients who are at greatest risk of this liver disease. And for some reason, we can also identify patients who seem to be almost immune to the liver disease. It's almost as if it is risk factors for this liver disease operating in a susceptible patient. And we've never understood what that susceptibility is. Simon Kokoschis spoke of the premature neonate having a state of physiologic cholestasis. We did some work with Dan Teitelbaum and using his mouse model. And then we did some work in fetal mice showing that the bile acid transporters that are impacted seemingly by phytosterols really are not even fully expressed in prematurity. So if you take fetal mice, these bile acid transporters are actually not adequately expressed. So I think there is a molecular basis for this susceptibility in the premature neonate. We know if you have a premature neonate with sepsis or a pro-inflammatory neonatal or perinatal condition, and no bowel, you can't feed, that child is very, very high risk for liver disease. In contrast, you have a term baby, you can feed some, and you need to give some supplemental parental nutrition, and the baby doesn't have comorbidities and does not have line sepsis every 2 weeks. This baby is relatively immune from liver disease. So I think we do have a very strong sense of what are modifiable factors, what are risk factors. And I think the attention on lipids should not be viewed as anybody saying it is lipids and nothing else. Lipids is one of the few, I think, one of the low-hanging fruits.
So what all other interventions do you think we could use to, or you are currently using in your clinical practice, other than, of course, enteral nutrition that Dr. Koran pointed out, that you're employing to minimize this effect of PNALD?
I can go first. Sorry, Dr. Ayar, do you want to go first, please?
No, you first.
No. So, but we have a fairly busy intestinal rehabilitation program. So what we are trying to do now is to look at these modifiable variables in the care of these vulnerable infants and see how we can improve the outcomes. So, for example, for those babies who have intestinal discontinuity with mucus fistula, how can we optimize their nutrition prior to reanastomosis? So we have looked at, to be able to feed as aggressively and as well tolerated as we can to get on fairly moderate amounts of enteral nutrition. And we also learn about the same things which we have learned. If an ostomy is more distal, babies get better. If they have weakened ileocecal valve or colon, that is much better. And with the practices like mucus fistulary feeding, once again, the outcomes might be better. And we are also looking at the other variables. What are the variables at the time of the reanastomosis which can make it better? Meaning, right now, our surgeons prefer to wait for 6 weeks before reanastomosing before reestablishing the continuity. If our preliminary data have shown that the longer you wait, the poorer is the outcome. So what is the optimum time? Is there an inflection point where after which if you wait long enough, the outcomes can be worse? So we are looking at nutritional and non-nutritional variables, which can perhaps improve the outcomes in these babies.
I'll go next. Really, Dr. Prem Kumar has hit the nail on the head. You've really covered all the points. I can tell you, Dr. Koran and I work together. There are others who know me. In my book, really, for the intestinal failure patient, there is almost no place for stomas. There is almost no place for stomas. And there is no value in waiting other than kicking a can that you don't like down the road in the hope that it'll go away. So it's in my book, that's counterintuitive to have usable bowel downstream and then say my child or my patient has intestinal failure. So that bowel should be recruited at the earliest opportunity possible, and should, if at all possible, even avoid creation of stomas in the first place. And only you and I can talk about this offline after if there's interest. So that's number one. So aggressive surgical recruitment of bowel, optimization of bowel function is number one. Number two, we've heard repeatedly about the importance of sepsis. Sepsis is a second hit. Sepsis is an independent risk factor for liver disease. Effect of endotoxin infused portally in experimental models. There are any number of examples of sepsis in the context of PNALD. So really, you have to look hard, and sometimes look hard repeatedly, for occult sepsis in these patients and treat it hard. There is never a contaminant in these patients. Every isolate is a real isolate and should be treated. And if you don't have explanation for a sudden rise in bilirubin, it's easy to label it as TPN and throw your hands up in the air. The search should be for sepsis then. If the child was on TPN for 4 weeks and nothing happened, and suddenly the bilirubin went up to 5 from 1, the answer is usually sepsis. And then feed. I think it really comes down to 3 simple modifiable factors.
Thank you, Dr. Iyer. That was
Yeah, I would just comment that I think besides some of these, what should maybe be first-line modifiable factors that are more simple to approach from a therapeutic or pharmacological approach, I think now that we understand some of the molecular basis for the defects that lead to cholestasis and phytosterolemia that are related to these nuclear receptors. Now we have some targets that are druggable, and there are drugs out there for both FXR and also some for LRH1 now that was shown recently in the paper by Ron Sokol's group using a phospholipid analog. So we are in the process, and we have evidence that, and there is some evidence in the literature that giving FXR agonists, bile acid and non-bile acid forms, are ways to activate FXR in the liver. And the idea would be that would be the expectation that it would upregulate some of these transporters that we know are effective, namely BSEP and the ABCG5G8 transporters. And so I think there may be therapeutic options here that might be worth considering down the line.
I would only say that, yes, there's a lot of pharmacological potential approaches, but if inflammation is the root cause, which it is, depending on the formulation or the condition, I think that, that there is a reason that EPA and DHA do offer an advantage that the others don't. It's not just reduction in omega-6 fatty acids, but you're now supplementing. For example, for linoleic acid, you would normally get your prostaglandin 2 series and your leukotriene B4 series. By using the omega-3 fatty acids, the real ones, the true ones, they will modify that. You'll begin with EPA, you'll alter it from the 2 series to the 3 series. And for the leukotrienes, you'll go from the 4 to the 5 series. So that's a general way to address the systemic inflammatory response. Then if you want to go from there to tailor make it with other pharmacological approaches that are very specific, as Dr. Burrin mentioned, and Dr. Iyer, that's certainly something to add on, but you may have the basic ingredients there to deal with this, since inflammation is at the core of this problem.
Dr. Karan? Dr. Valerie Karan.
Sorry. The only other thing that I was going to add, as we were talking about the modifiable risk factors, and I mentioned feeding also, this is not a drug, but breast milk, encouraging families to breastfeed whenever possible. We do know it's maybe not be possible for all families, but there are a number of hormonal factors in breast milk, including GLP-2, that have been shown to enhance adaptation. And so I think, although there's no large RCT that we've seen so far, but there is some retrospective data that suggests that using breast milk and also perhaps some elemental formulas would be beneficial in helping with adaptation for these patients.
Thank you. Yes.
Sumita, can I make a quick
Yes,
comment
please.
if you have time? We're talking about modifiable factors, and one of the modifiable factors, so we recently did a national survey on the utilization of lipids. It's going to be published very soon, I think in the next month or so. It's in the second revision. So what we found that really interesting, because we had around over 100 centers, level 3 and level 4 neonatal intensive care units, and the use of availability and utilization of SMOF has overtaken that of intralipid. This was just a snapshot. We wanted to capture it because if you don't capture it now, you will lose it. So, 10, 15 years ago, we did not have SMOF on our formularies. We are not using, and we are using it now. I would like to say without much thought. And 1 in 5 centers did not have a lipid protocol, and they were trying to make these switches. And then we are not thinking about our patients. And the other worrying thing was dose limitation, which has been shown, which a lot of caution has been raised with dose limitation with SMOF was being practiced by, a significant proportion of units. And what I'm trying to highlight here is there is a lot of heterogeneity. And I think if we try to standardize our protocols, and I think that will be a good way to improve outcomes. And that's one of the reasons why in studies like, the Dr. Westpal mentioned, where the outcome, which was the bilirubin, was so low. So in some of these studies, when we standardize our protocols, the outcomes get better. I think there's a lot of power in standardizing our regimens, and we can improve the outcomes. That's a very underutilized tool, I think.
Thank you. Those are very thoughtful comments, and we really look forward to your publication, because that is going to be very, very informative and helpful. Does anyone have any last few comments that they would like to make before we close the session?
No, I had another comment. Sorry, I know we talked about the difficulties in getting the terminologies. I know intestinal failure-associated liver disease has been the most acceptable, but I find it even that suboptimal because, first you have to define intestinal failure, and then you have to define intestinal failure-associated liver disease. And we say intestinal failure is dependence of parenteral nutrition of 60 out of 74 days, but we see liver disease as early as 2 weeks. How can we make a diagnosis of intestinal failure-associated liver disease even before we make a diagnosis of intestinal failure? I'm just highlighting the difficulties in capturing this definition. I don't have any viable solutions, but I would like to see the thoughts of the panel.
Ruby, if I may respond.
Yes, please.
I don't have a good response, but I wanted to comment nonetheless. One, I clung with increasing difficulty to the term PNALD rather than IFALD, because at least in whatever now 25 plus years of looking after intestinal failure patients, I'm yet to see a patient with limited bowel length who perhaps needs IV fluids alone and not parenteral nutrition, as most of us would recognize it, develop liver disease. The syndrome of liver disease that we're talking about today occurs only in patients with intestinal failure if they are getting parenteral nutrition, as most of us would agree. If you have a patient with short bowel who needs, let's say, intravenous magnesium once a week, intravenous fluids once a week, and that type of patient exists and has intestinal failure by definition, I would invite anybody on this panel or in the audience to report their experience of liver disease in that cohort of patients. I submit that doesn't exist. Therefore, intestinal failure-associated liver disease as a term has been a poor term. I suspect more to do with our frustration in not making progress with the disease than with any meaningful contribution to science. But my comment to Dr. Prem Kumar is, I think you brought up the issue of surrogate endpoints more than once. I think it's a valid point. It's an important suggestion, but it's hard to know what are meaningful and actionable endpoints.
One thing I will say, and I think I agree with you, you commented on this earlier, and many people have shown this, that hyperbilirubinemia in these patients is a very strong predictor of mortality. Multiple studies have shown this, and time again, we get patients referred with a high bilirubin, and the referral was an afterthought. In our hands, if somebody's bilirubin is starting to rise in the setting of intestinal failure, that's time for a full court press. That the horse is kicking at the stable door, and we need all hands on deck to turn this around. So hyperbilirubinemia is a late phenomenon, waiting for loss of synthetic function, as we see in other liver diseases, the high INR, the falling albumin, or indeed esophageal varices, which almost never occur in the setting of short bowel. Those are late stages, and it's too late. We need early surrogates. So Murli, what are the surrogates?
I don't have any bright ideas, but I think nutritional outcomes, for example, growth outcomes, are more meaningful, and even tolerance of feeds in terms of volume or calories can be viable endpoints when trying to compare these different lipid emulsions.
Does anyone have any other last comments? I guess we had a very lovely and lively conversation. So thank you all for the lively discussion this afternoon. Dr. Judy Racusin will now rejoin us for a wrap-up, and thank you.
Good afternoon. This has been an incredibly productive meeting. I really can't thank the panelists and participants, speakers enough. And I'll get to some thank yous in a moment. But first, I want to wrap up what we heard today. This morning, we heard a lot about where the incidence of PNALD IFALD has gone over the last 20 years, and we've seen that it's declined substantially. However, it's very clear that it's still an issue and something that we should, continue to take seriously. With the current management, in the cases where it's being well managed, it seems to be less of a problem in neonates and younger children, but obviously, as we've discussed, needs to be continued to be monitored for. And in adults seems to be still of a significant concern and something that needs attention to. As we're managing these patients, we need to balance between the need to prevent PNALD IFALD, but also not letting optimal patient management be driven by, I think, what we heard this morning called a fear of PNALD, and so that's something to keep in mind. We also heard that the nutritional needs of preterm neonates are different than older children and adults, and that this population may well benefit from, I think we might have heard the term designer lipid, and we hope that people listening and participating today will take on that challenge. We also heard that that phytosterols, and we heard this morning and this afternoon, are just one component of the PNALD IFALD issue, and that there are a lot of other places that need attention. We heard this morning also that the fibrosis may not be reversible that occurs in PNALD IFALD, even though the signs of cholestasis, the biochemical measurements may go away. And then lastly, from the morning session, we heard very clearly that there's a substantial need for better training in parenteral nutrition, and as was echoed this afternoon, maybe particularly in adults, but all across all across our patient population, from the youngest to the oldest. With regard to this afternoon, I just want to touch on the questions, and I think that we did hear some evidence to support, and I think the panel reflected on this, that phytosterols are a dose-related hepatotoxin. And then the question of whether a phytosterol-depleted lipid formulations are scientifically feasible, I think we heard very clearly today that there are unintended consequences of using large industrial methods to try and deplete phytosterols in the sense of removing other compounds within the lipids that are beneficial, but also not knowing what other compounds are created in that process that might, be potentially harmful. We did talk some about clinical trials and the challenges of those. We heard, we know and we presented this today that in the PRIA PMR that Fresenius Kabi did comparing sophlipid to intralipid with the PNAC outcome, that we had many fewer cases than anticipated. And we would anticipate, powering these trials to continue to be challenging. I think that that the discussion of the challenge of clinical trials kind of morphed into a discussion of real-world evidence, and we totally, hear you. Heard our speakers this afternoon that these are outcomes of intestinal failure and IFALD, PNALD, IFALD, that it's challenging to look in real world data for these outcomes when there aren't ICD-9 codes to correspond. We also know from our own experience at FDA that when looking in these large data sets, it's often very difficult to determine what intravenous lipid emulsion a patient actually got. So the way that things are tracked in these databases, we know that a patient got an intravenous lipid emulsion, but not which one, which obviously would make it difficult to make any kind of comparisons between products. We heard a lot about other modifiable risk factors this afternoon, and importantly, the emphasis on prevention rather than treatment, and really emphasizing and encouraging opportunities to feed infants, including with breast milk. And in addition to the feeding strategies, aggressive surgical strategies to recruit bowel, aggressive treatment of sepsis, prevention and management, and looking for those occult sources of infection. And then I think finally we heard about some new ideas for therapeutic targets, which we will take under consideration. So with that, let me turn to the thank yous that I have today. Again, I want to start by thanking our speakers and panelists who've taken time away from their patient care responsibilities and their labs to share their expertise with us today. We really, it's been tremendously gratifying that with the level of interest from our speakers and panelists, and we really appreciate your efforts. Second, many thanks to Brenda Stoddart and the staff of the Small Business and Industry Assistance Team in the Division of Drug Information for organizing this workshop, keeping us all on track. To Barbara, Mike, Chris, and all of the staff here at the CDRH television studios, we could not have made this meeting happen today without you. I want to thank my colleagues in the Division of Hepatology and Nutrition, as well as those in the Office of Pediatric Therapeutics and the Division of Pediatric and Maternal Health for their assistance and support in putting on this workshop. We appreciate the participation of representatives from Fresenius Kabi and Baxter for sharing their perspectives. Finally, it's always patients and their families that we have in mind when we hold a meeting such as this. For patients of all ages who need to be fed with intravenous lipid emulsions, we're aiming to develop strategies that will improve health and minimize risk. We intend to use what we learned today to benefit you. Have a good weekend, everyone.